rupees to set up a decent mass spec lab, something around 15 crore to set up a complete lab that you would have good instrumentation, all the kinds of protein cleanup facilities, standby liquid chromatograph, sometimes a standby mass spec itself. So things cost a lot of money. And then in any translational work, you would like to take your solution right from the lab to the clinic, from the lab to the agricultural land, right? That is what a good translational work would be. Like, it's a test that can be done by a layman, a test that can be as easy as like those lateral flow tests that you use, like for COVID we had used one, pregnancy testing kits. So something as easy that can be done at home or maybe done at a very rural dispensary. That's what we aim at as a scientist. So one way of doing it is by microarrays. Now microarrays, I think the first thing that reminds you about a microarray is that it is something related to DNA. And of course it did start off with DNAs, but now we have all kinds of protein microarrays, and that's what we will be discussing. And we will also telling you, using a case study from the work that we did at IIT Bombay while I was there, at how you can use microarray as a diagnostic and prognostic tool. So if I ask you what are microarrays, what would be your answer? Anyone wants to volunteer? What is a microarray? What is micro? Small, right? We have microscope, so all kinds of micros we have today, micro USB. So micro means small. And what is an array? Collection of
instances
of what?
Particular specific object, or you can say protein, genes,
right, anything that is systematically arranged in a repeated manner, right? That's an array. So if you go to the definition, so these are very miniaturized platform, and what protein microarrays can, or any microarray can do, it can allow for simultaneous detection of molecular interactions, right? Here the word is molecular interaction. It can be gene expression. I think Professor Graham started off his AI ML lecture with showing you a microarray grid, and he will be also going deeper into it sometime soon. And so it can check for gene expression. It can also check for protein binding. And what you can do it, you can do it on a very small, I think all of you have seen a microscope slide, right? And it's very small, it's very handy, you can carry it with a hand. That makes microarray something which is extremely portable. You can take it in your pocket, and the work that you need to do is, it can be done very fast. So what typically a microarray will consist will be an arrayed series of microscopic spots, and each spot will contain a specific molecule. It can be any molecule. It can be DNA, it can be RNA, it can be protein, it can be cell lysate. And what you do it is you immobilize it so that it does not move around on a surface. Usually it can be a glass chip, it can also be a silicone chip. And it all started with people started designing microarrays of DNAs, and what they did was they put small fragments of genes, DNA, and they put it on a glass chip, and when these spots were exposed to a sample containing DNA or RNA, they would hybridize, not bind. So DNA RNA will always hybridize, and this hybridization would lead to a generation of a signal, and based on signal intensity and the presence or absence of signal, you get an expression data set, right? So what you do is you can compare different types of RNA from different samples. You can healthy versus diseased can be one way. Like you have the RNA, entire RNA you spot from a cancer tissue, from a non-cancerous tissue, and that will give you at the level of transcript, that is, it will tell you what are the genes overexpressed and not expressed or underexpressed. One thing you need to remember, these are always targeted assays. These are not by far discovery. Why? Because in most cases, you know what is what in the array. So this is one challenge of a microarray, that you cannot do much of discovery work, except for a few things that we will understand very soon. It can also, you can find single nucleotide polymorphisms if you're working with DNA. So these kind of stuff you can do. You can compare the genomic hybridization, you can compare the number of genes in a cell, and you can visualize the DNA, the interactions, because most of these probes will carry some fluorescence dyes tagged to it, Psi3, Psi5, Alexa fluors, and when you scan it, you get a signal, right? Red, green, blue dots. So why do we use it? Because we can use it to understand diseases, drug responses, developmental stages. We can also study various mutations. We can also identify genes or proteins linked with a specific condition. So these are the things that you can do with the microarray. But when we talk of protein microarrays, so this was a later development, and what people did was similar to DNA microarrays. They started printing small proteins onto a surface or a glass chip, right? And these proteins can interact with other proteins. And how these proteins interact? A very common interaction is antigen-antibody, right? So if you have antigens presented on a glass chip, antibodies will come and bind. You can do the reverse. You can print antibodies on a glass chip, and then you would see what are the antigens that are binding to it, and that can give you a protein profile. It can also give a functional analysis, and you can do a kind of a drug discovery approach. You can do all these stuff. And this figure, I don't know how, is it visible well, or should we, can somebody turn off that light? This one? Right. So I think it is visible to you. Right? It's fine. So these are grids. These are small grids. And if you look at it, these are very small dots present on this microarrays. And each of these dots might represent a protein or a peptide, or sometimes a cell lysate. So depending on the kind of arrays. So we will now move on to what are the major types of protein arrays. So the major types are primarily these. So these are analytical protein arrays that are used for, so these are all analytical arrays. So what you have antibody arrays, you have peptide arrays, you have antigen arrays. We'll talk about peptide and antigen very soon. But what is an antigen array, antibody array? You have, imagine you want to check a cell signaling pathway, and you want to check that for a particular pathway in the developmental stage of my animal model or cancer, you want to check all inflammatory markers. So what you can do is you can print all the antibodies against inflammatory panels. Maybe you have a panel of around 50 or 60 markers. If you do, and you have to screen perhaps hundreds of samples. Now think that each grid contains 60 spots, and each of these spots in duplicate, that means a grid of 120 spots. Each of these duplicate spots are primarily an antibody against a specific protein in that cascade of signaling. Now you take the entire protein cell lysate, you incubate this glass chip. In one go, you can do a sort of an ELISA or Western blot of all these arrays, of all these proteins. If you have to do these individually, that you have to do for these 60 proteins, it will be extremely costly, and forget costly, it might be extremely laborious, and then you would have variations, because perhaps for these 60 blots, it would take at least 4 to 5 months. Temperature conditions might change, your mode might change, your buffers might change, and the lysate that you took, that after repeated freeze-thaw cycles, even that condition might change, right? So you can do it in one go, in one shot. Then you have peptide arrays, these peptide arrays. Now what are peptides? Short fragments of proteins. Now suppose you want to check that if your protein of interest is trying to interact with another protein, right? You have some proteins, you want to do a protein-protein interaction study, right? So you can either do a peptide array, or perhaps you want to check for antibodies present against a specific protein, whether you are generating autoantibodies. Now what you can do is you print these peptides, and almost similar is antigen, you can print it onto a glass slide, and then you can do something that we will discuss later, something known as an epitope mapping. You can do an epitope mapping. You can exactly know which part of the proteome is actually being antigenic. So if you go back to your immunology basics, you will understand that the entire protein is never antigenic, right? There are specific regions in the protein that are antigenic, and we call it the epitope. Now if you want to design a vaccine candidate, suppose you want to design a vaccine candidate. This vaccine candidate cannot be the entire protein. Maybe you want to design a peptide vaccine. Because proteins by itself can break down, it is mostly always thermolabile. You would need a cold chain system to maintain that entire protein cells. But suppose you want to make a peptide vaccine that you can carry in a pocket. You can use it as a nasal spray. So in the nasal spray you have this peptide mix. Because they are peptides, they are far more robust, and all you need to develop or to administer a vaccine is just do 2 sprays in your nostrils, and those peptides will be absorbed in your nasal mucosa and will start generating an antibody response. But to select for those epitopes, you can use these arrays. Now another kind of array is called the functional protein array. This is actually very important when you want to study protein-protein interaction. You can also do an enzyme array. Suppose you want to check for enzymatic activity. What all enzymes are present in your cell lysate? Then again, you cannot store your cell lysate for a longer duration. Why don't you go for an enzymatic array where you can have substrates, and each of these substrates are unique to the enzyme, and if the substrate reacts, it will create a chromogenic spot. And based on the presence and absence of the enzyme, or maybe based on the presence or absence of the activity of the enzyme, your spots will change color, maybe something from faint yellow to brilliant yellow. And these arrays can again be scanned, it can be digitized, and once it is digitized, you have Professor Ball who will help you to apply his magic of AI and ML to understand your data better. But you can do this for a lot number. See, the arrays help you in increasing the throughput of your experiment. Now you are not looking at one single cell or one single protein, or you can look at the entire cascade. You want to study glycolysis, and you can have all the enzymes that are involved in glycolysis. In fact, you can go for some secondary enzymes that are part of the upstream and downstream cycles, and you can have a complete understanding of how these cycles are processing going ahead. Something that we do is reverse phase. These are all normal phase, and just as in HPLC or chromatography, reverse phase means here you put the cell lysate or the tissue lysate onto the glass plot. Right? Now suppose you have to check 500 samples for the presence of one antibody, and you want to do it very fast. You are in a rural clinic where there is a breakout of a pandemic, and you do not have the money or the time to give 500 results. You take a glass chip, you take a microarray printer with you, you take their blood serum samples, spot it on the glass slide, and then do one check, and based on the arrays, it can tell for those 500 patients at one given time if they're having a specific antibody, or if you are checking for a specific biomarker. So these are called reverse phase arrays. So now just going deeper how this analytical microarray works. So how they work is, these microarrays are designed to capture a specific protein and then detect their presence. I think many of you would have heard of would understood something known as ELISA, right? It's something of a micro ELISA, that we have miniaturized on a glass slide. So what they do is, if you have an analyte labeled, so this is the antibody, and this is the fluorophore. Now, and these are the reporter antibodies, and this hexagon is the target protein. Now, this target protein will bind to it, and the concept of sandwich ELISA is what? That if the target protein is binding, then it would not allow the reporter antibody to bind. So a binding event will be distinguished by the loss of signal. So if you have a bright spot, means there was absence of the target protein, right? If you have a really bright spot, because reporter antibody could bind with this antibody, and because target protein was missing. Now if you have the target protein, it will sterically hinder the binding of the reporter, and then you would have a very low faint signal. So faint signal means your target protein was present. A bright signal means the faint signal was if it is a bright spot, means your targeted protein was absent. So this is how analytical microarrays work, and these are specifically designed to capture specific proteins, detect their presence, and also sometimes, if you can standardize it well, it can give you a concentration in a given sample. So this is again something that is subject to specific conditions about how you have done the study. And they usually contain antibodies, aptamers, and other protein binding molecules. And why they are used? Disease diagnosis. We have just discussed that you go to a rural village and you want to diagnose 500 villages. You want to do a sero survey for all the 500 villages in one go. You can do it rapidly at a micro scale at very low cost, because since you have scaled down the experiment, your consumable cost will also be reduced. So in India specifically, because most of you, I think all of you are from India. So one thing, always remember, in India, when you develop a technology, make sure it's cheap, else you cannot translate it. We are not a first world country who can afford to have a 5,000 or 8,000 rupees test. So ideally you should aim something between 10 rupees and 100 rupees. Each test should not cost more than that if you really wanted to translate it on a big scale. Also, you can do protein profiling. If you're not working on diagnostics, you want to work on your own cell cycle systems or your disease models, then you can check for the relative abundance of various proteins in different samples. That also gives, without using a mass spec, a fairly good picture of the biological process. One thing I think all of you must have understood by now, nowhere did I mention that you have to deplete your sample. Right? You don't have to deplete your sample. The sample processing step in these microarrays is minimal. You have to just take, if you're working with blood, and we have worked for COVID, all we took was, we took the plasma, or rather we took the serum, and we diluted it 100 times, 200 times. So even if you have to take blood, you don't need to draw 5 ml or 10 ml. Even 1 ml draw would be sufficient for maybe 200 microarray experiments. So that way your procedure, or your test is cheap, is non-invasive or minimally invasive, as well as translatable to at a very low cost. Then we have something known as functional microarrays, and as we had discussed, they contain full-length proteins or protein domains or peptides, right? So if you're wanting to study a protein-protein interaction, or suppose you want to study if a particular domain of a protein is interacting, then you can break down the protein, you can make those proteins, immobilize it on a chip, and then do your assay. And each different domains will give you different signals using the same sandwich principle, right? So this again gives you much more flexibility to work, and it can be used for protein-protein interaction study. You can also do protein-DNA RNA interaction study. Now, suppose you are working on a transcription factor, right? And you want to check, or you're trying to do a promoter analysis, and you want to check whether my specific. So these kind of proteomics technologies gives you. This is not an alternative technology. This is actually a technology in its own right, and it gives you much more advantage compared to a mass spec, because mass spec can never tell you if a protein or DNA is interacting with each other. It can predict, but it cannot give you hard evidence. You can also test potential drugs, whether a drug is binding to the protein of your interest, and it can also provide some actions, like it can give you some action points that how if the binding is being there or not, which domain is binding. All these kinds of studies you can do with a functional microarray. So just to tell you what kind of functional microarrays we have a protein-protein interaction microarray. We have a protein-lipid. P shows protein-lipid. C shows protein and cell lysate. D shows small molecule binding. E shows substrate identification of PTMs, substrate enzymes. Then you have protein DNA, protein RNA, antibody specificity or serum profiling. So you see just one technology, but with the variations it can provide. And if you see, none of these studies could be done on a mass spec, right? And they are usually very cheaper. So again, to get into details of reverse phase protein arrays, as we had discussed in the very introduction, that reverse phase is when you spot your cell lysate and then probe for a specific antibody to detect the presence of a target protein. Now you have 1,000 samples. You are working on a clinical, you are trying to establish a biomarker. Suppose you have established a biomarker using mass spec. Fine. You have established a biomarker against mass spec, and you are telling that this protein should ideally be a prognostic tool or diagnostic tool for disease X. Now, one good way of, you don't have, now you want to try to reach out to your collaborators across India, across the globe, and you want to validate it. Now you cannot ask everybody for a sample and they would ship it back to your lab in India, and you will run thousands of targeted proteomic assays. What you can do rather, you can do a reverse phase. You can tell them that, okay, you print it on your chip, and this is the protein antibody I'm providing. So if this antibody is binding to your serum, means this specific protein has gone up or is present in the serum or plasma of the patient. That can be you can validate your biomarker. So sometimes you can use these techniques to validate the findings that you get on a mass spec. So of course, disease progression is one of the ways you can do that. You can also study disease progression. Suppose there are stages of cancer, stage 1, stage 2, stage 3, stage 4. Then you have diseases like COVID that has a non-severe and severe stage. All these things you can do. You can also study the signaling pathways in cells. You can study post-translational modifications. So, and then how does it look like? So, all you do is you take samples and tissues, lysates or serum or body fluids, or also subcellular fraction. You make a homogenate, and since these are proteins, it won't usually stick to a glass slide. You can take a nitrocellulose membrane, and on those nitrocellulose membrane, you can just allow these molecules to be absorbed in a particular array pattern. One thing to remember is, whenever you design an array, you have to remember which spot you are putting what. So on your computer, you should have designed the array, and just like you work in a 96 well format, you don't label each of the wells, do you? Right? You know that A1 is this, A2 is this, B3 is this. So similarly, think of it like a 96 well. It's a 96 spot array on a microscope slide. Then you can probe with fluorescence labeled antibodies against these biomarkers. And one more thing that you can do, you can actually do a simultaneous detection. You can use 2 antibodies. So 3 antibodies, depends on the amount of money you have. And also, when I showed you the first grid, now this is here also you have a grid. So sometimes you can do specific incubations on specific grids. So the first grid you can incubate with antibody 1, second grid you can incubate, and there are 8 grids and 16 grids that can be possible on a specific microscopic slide. And based on the grids, if you have 16 grids, in one go you can assay for 16 different antibodies. If you have 2 types of fluorescence markers, you can go for till 32 proteins. So this is, since we are talking of big data, these are actually more of a high throughput approach that you need to think. So we don't talk in a high throughput approach, we don't talk about one data set or 2 data points. The data points should be large. So here the patient number will be large, the number of samples, assays you're doing should be greater. Now, one interesting thing is the peptide microarray. So here what you do is, instead of full proteins, you can print small sections of the protein and peptides, and since you're printing specific regions of the peptides, each region of the peptide will correspond to a specific motif or domain in the actual protein. And the number of applications that you can do is, one is epitope mapping, which we will discuss very soon, and which was much of my work that I did at IIT Bombay. So we'll discuss this in detail. And you can also study how specific modifications like phosphorylations on a peptide might affect its interaction. So suppose you have a protein that can get phosphorylated at 12 different positions. And yesterday I think we were discussing that different phosphorylations might lead to different kinds of activity. So you can have all the permutation combinations of those domains with different phosphorylation in each domain, and you can try to understand how each phosphorylation is affecting the biology of the protein. And how do you, what do you do? First of all, we can do biomarker detection. You can presence of biomarkers. You can have autoantibodies or autoimmune disorders. Very easily you can screen. Then you can do autoantibody profiling. Again, for autoantibodies, indicating autoimmune disorders, you can check for those profiles that, okay, these particular person is having autoantibody against these proteins. You can do viral and bacterial detection. We will discuss how we can detect COVID, and you can do epitope mapping. Also, you can do drug target identification. You can find, okay, this is the drug that interacts with this domain or motif of the protein. You can personalize treatments based on these profiles. And also you can screen a lot number of drugs using this, because you are using a high throughput approach. You can screen multiple drugs, how they're interacting. So multiple studies, you can do interaction studies, you can do post-translational modification studies. You can also, as we discussed earlier, you can also try to find what are the probable or the good vaccine candidates, right? So now we will see how we used this microarray while we were doing our studies on COVID-19 in India. So this is the general trajectory of what happened during COVID. So what happened was viral particles entered into our airways, and the presence of SARS-CoV-2 receptors, the ACE2 receptors was actually the ACE2 protein that was present on the cell cycle, on the surface of the lungs. They allowed the virus to enter so they allowed the protein to interact, this virus to interact, and they got internalized. Then the mumbo jumbo of immunology, we had the antigen presenting cells and the helper T cells. It helped and activate, and the macrophages and the dendritic cells, all of them together led to the activation of the helper T cell, which activated the B cell. This B cell started producing the plasma cell. This plasma cell again produced antibodies, and of course there is a class switching event. So initially they produced IgM or IgA, and then they produced IgG. Now what we could do is, using these epitopes printed, or these peptides printed onto the glass slide, we can see which peptides were actually antigenic. The entire virus, or just like any other protein, the entire protein can never be antigenic. So what did we do? So this was something that we purchased from a company called Pepperprint in Germany. So what they did was they printed all the peptides present based on the Wuhan original sequence that was sequenced at Wuhan in China. They translated into the viral proteome, and the translated proteins were printed in an overlapping manner. You see, the first amino acid was 1 to 15. The second was from 3 to 17. So there was a 13 amino acid overlap. So now you're creating a grid where each of the peptide has a 13 amino acid overlap. So I want you to visualize, if you do this overlap, you have a very high resolution of it around 2 amino acids. The resolution that you're achieving is of 2 amino acids. 15 amino acid peptide, and you have a 13 amino acid overlap, which means this grid is actually moving 2 places at a time. And all the peptides right from nucleoprotein, spike protein, ORF3a, envelope, membrane, ORF1ab, ORF6, 7a, 8, 10, all these were printed onto a glass slide, and that led to 4,883 spots multiplied by 2, because all these spots were in duplicate. So these number of peptides were printed onto one glass slide. Of course, it came at a cost. It costs us around 65,000 rupees for one slide. But what we did was, we wanted to ask a biological question that does severe and non-severe patient have a differential expression? Does their immune response vary? Right? So what is it that few patients getting infected by the same coronavirus is having a very extreme severe response, somebody is having just a normal response, usual response. So is it their immune response in the first place? Is it how their immune system was latching onto the peptides due to neutralizing antibody response or binding antibody response? So all this we wanted to study, and for that we will go to the linear. So what we did was a linear epitope mapping. So this is a linear epitope mapping, and if you clearly see here, so these are overlapping peptides, right? So this was the entire SARS-CoV-2 protein, and you see there is a fair amount of overlap by the way the grids are kept. These overlapping peptides were printed onto a glass chip, and the IgG or IgM as possible were started to bind. Now imagine a specific antibody that is binding from, suppose antibody, its binding site is between amino acid 4 to amino acid 18. That is the motive. So it will very weakly, but surely it will bind with the first, because there is an overlap. It will also bind with the second, because there is an overlap. Now this binding will create an array of spots, right? And since we have printed in duplicate, it will be a double spot. When there is a double spot, you would, via data analysis, we could actually look into the specific consensus motif. The specific consensus motif. So this is the binding pattern, and this binding pattern will help me identify with a high resolution of only 2 amino acids. Only 2 amino acids. This will give me a very high resolution about, okay, this is where the protein, the antibody is binding. It can be IgG, it can be IgM, it can be IgA, depending at what you want to study, right? So this is what the grid of IgA used to look like. Now you see, this paper is already published, so in 2023. So this was what the scan looked like. Now, if you look very carefully, you will see the entire slide is not stained red. You can see small dots, right? So these are all double spots, and when you zoom in further, so this is the overlay of the grid. So the computer, so the guys who have printed this, they already know which peptide is present at which spot, right? When we overlay the scan with this grid, we know the signal is coming from which peptide. And based on that, what we found, if you look at it, I don't know if it is visible, so this is the severe group, the severe patient. This is the non-severe patient. Now in severe, you find a very strong expression, whereas in the non-severe group, you find a very weak expression pattern. This is just a representative, and this is how we plotted it on a slide. Similarly, again so you find the non-severe, the severe group, at least in this IgA channel, initially mounts a very strong antibody response against ORF1ab and these specific regions of the protein, right? So these are ORF1ab protein, this is also ORF1ab, and I think this is the glycoprotein, membrane glycoprotein, this is also ORF1ab protein. You can go to this paper and you have all the data sets available. So this is how we wanted to understand if there is a we could distinguish the response given by severe and non-severe patient. Thereafter, we moved on to the IgG response. And here also, we can see that in case of severe, and this is this classical double spot. You see, I think this is very clear. So this is a very strong binding. This is for nucleocapsid, this is ORF1ab polyprotein, this is for nucleocapsid, this is for ORF8 protein. So again, you can see this grid. So this entire slide did not have this green background. So these are specific spots, and all of these spots, if you go back to the literature of the manufacturer, we can exactly tell what these spots are. And what you see in these boundaries is the control, and what could possibly be the control. So this checks for polio. It's against polio virus, and all of us are vaccinated against polio. So we would have a strong response for polio for all the patients, but this is only for SARS-CoV-2 proteome. So this gives you an idea about how you can use microarray. So I told you about different forms of a microarray, and I also showed you how you can, so these targets, these are these protein targets, we can easily say, if we suppose take this protein target and we tell that if there is antibody response against this specific peptide, a clinic can usually do an ELISA, right? We can convert it into an ELISA target. A clinic can also run a microarray, but perhaps they don't have a scanner. Microarray is also costly. Slides cost 50,000, and the scanner that we used was around 2.5 crores. So yeah, omics is actually costly, because that's mostly because there is very less adoption of the technology. More and more, you go back to your labs, you convince your supervisor, and you have more sales, the prices come down. So that brings us to the completion. So you can just go through this paper. I will share this paper at the end of, as I said, few of the papers that we have been referring to. I will also ask for some papers from Professor Graham Ball, some reading material that you can read through. Right now, if I even share the reading material, you hardly have the time, because we are moving from one topic to the other. So once the paper is done, I mean your entire course is done, you can go through these papers, these folders, study material on microarray, study material on targeted proteomics, study material on MetaboAnalyst. So all these tools that we are discussing with you, Reactome, MetaboAnalyst, they have their own individual research papers. These are developed by research groups. You can go back to those papers, read them. Most of these tools would also have their own tutorials that you can actually spend 4 to 5 days. So there are Max Quant summer schools. I think that runs for almost 5 days. So of course, whatever they will teach in 5 days, we could hardly cover in 1 hour. So the idea is to give you a flavor. things exist. You know MaxQuant exists. It's free. You can use it in your laptop. Perhaps if it is having a 16 GB RAM, but mostly you don't have to pay money for it. So that brings us to the end of this session. If you have any questions, and this is why you should attend this GIAN program. So the fire needs to be kindled, right? We are not aiming to fill you with information. We'll give you more and more things to choose from, and then whatever you feel like you want to carry on forward in your own research, in your area or domain of expertise, pick and choose. So anybody has any questions? I think we can take a couple of questions at most, and then I will hand over the stage to Professor Graham so that he can continue the proceedings for the day. If you have no questions, I feel you have not understood anything.
Sir, most of the applications that we told us about in the disease identification or drug delivery.
Right.
So, don't they have any application in plant and animal studies? And
Plant and?
animal studies. Effects on diseases and health, I would say.
As I said, when you study cell cycle, right, your plant would be having lots of, so those are most of these studies that since I have been working on animal models and diseases, and so I picked up examples. Of course, they would have it for plants. So plants would produce proteins, and proteins would obviously react with antibodies, but using those you can make your own arrays. I mean, there are companies like Pepperprint and Ray Biotech. I think in India you don't have it. You have to go abroad. You have to talk with them. They can make you custom chips, but you can do it. It's not impossible. If it is a protein or a peptide, it will definitely bind with someone or the other. You can do DNA protein interaction studies. So if I go back to this slide, so you see there are so many interactions, and this interaction can be translated to algae or plant or animal or to a disease. So it's up to you. So that's how you, as a scientist, you have to wonder what possible thing you can use. If you want to use as a protein lipid interaction, you can go for customized microarray printing. If you do it in bulk, of course it will be cheap. If you ask for one chip, of course it will be extremely costly. But things can be done. Anyone else?
So, what scanner exactly is used?
So there are arrays, scanners like GenePix is one of the scanners. You also have this scanner. This is from Inopsys. So the photograph is from Inopsys, their website. So Inopsys is one scanner. GenePix is one scanner. If I'm not wrong, Agilent also makes a scanner. So the reason they are very costly is because they use lasers that are these are monochromatic lasers. This is the laser I'm using is very cheap, because if you look at the wavelength, it might look green to you, but these are not perfectly monochromatic. So monochromatic lasers, one reason why they are costly. The camera that they use, you get extremely high resolution. You see, these are microscopic slides, but when you get the actual image, it's almost like 45 MB file, and you can zoom into 2-micron dot, and it appears like a circle or a bar. So that drives up the cost. But again, if you do the work in collaboration with a lab that already has a scanner, it brings the cost down, right? And in India, I think there are almost 5 or 6 scanners that I personally know. One is with Professor Sanjeeva, other is I think at NII Delhi. One is at Thesti Faridabad. So I'm not very sure about these things if they're still working. But when I was wondering, so you have one Agilent instrument in BHU itself. So you can do that. I mean, you have to plan your work. You can do your incubations, and you can just scan them. And these slides are very robust. Since it's a glass slide, mostly you can carry it in your pocket. It does not need a dry ice shipment. And most importantly, for human studies, you don't need to deplete your plasma sample. That's one of the big advantages, that you can save a lot of money on sample processing costs. No buffers, nothing. All you need is physiological saline and saline and few of these buffers that you usually use for Western blotting. A blocking agent. I did not go into the protocols, but we can share with you the protocol as well. This paper that I was mentioning, it has a protocol. Okay, anyone else? Or I can wind up and I can hand over the stage to Professor Graham. So thank you for your patient listening. Professor Graham, welcome back. The stage is yours.
Thanks.
It has inputs, has outputs, and has weights, just like any other neural network. The only thing is, there is a looping weight which takes a value from a previous time point. So instead of having an input, a lot of inputs and one output, you have one input and one output, but then that input and output moves over a time series. So let's say we have 10 values, T1, T2, T3, T4, T5, T6, and so on. At the initial time point, we just have a bias value. There's no input from a previous time point because there isn't one. At T2, you have an input that's coming from the previous time point and the current time point value, and then that predicts time point 3. So there is this chain of events that is predicted. So the idea is we look to capture patterns over time. So you can imagine how something like the dollar rupee might change over time. There is a pattern in that. So we know what happens at this time, we know what happens at this time. Based upon what's happened over the last 2 years, can we predict what's going to happen in the next week? That's a time series analysis. So basically for day X, can we predict values for day X plus 1, day X plus 2, day X plus 3? We're reading the future. So each day, what happens over time is encoded in a recurrent loop. It has information about what's happened in the past, that's stored in weights, but it then has an element of recurrence over time. So the weights, so variables on the previous days are used to predict subsequent days, and the weights are saved at each time point to capture that variation over time. So you have effectively a weights matrix which represents the variation over time, and that's rolled into the network. So what does that look like? Well, we have t minus 1. We have a bias, and we have a value at t0. That's input to produce an output. So we have an input, which is a value t minus 1, and we have an output, which is a value of t0. Yep. We have a weight, or at this point we have just a bias. What we then have is that the output from this model comes down to here and compares with what actually happened. And then we have that as an input, so the t0 is the input, t plus 1 is the output, plus it receives a weight from the previous link in the chain. So it's captured what happened before, and it's feeding what's happening into the next one. And so we have this sort of looping through the time point where an input becomes an output, weights are stored at each time point, and we effectively work our way through the time data. So if we have time data for 5 years, then we capture all the variations and patterns in that so that we can, if we have a particular set of events at this time, we can predict what's going to happen in the future. Now, that's a complicated view. What effectively we have, and this is called unrolling, so we're unrolling the network here, but we can roll that network up into a simplified diagram, which means we have an input, an output, plus a recurrent loop, which encodes the weights for previous time points. But you have to kind of unroll it to explain it, but normally in notation of neural networks, this is how it's represented. So if we zoom in to one of these blue boxes, what do we have? Well, we have the output from the previous node in the chain, which is effectively a weight, multiplied by a weight. We have an input, which is the next time point, multiplied by a weight. We pass that to a transfer function, and that goes through the algorithm to give us a new prediction. And each one of those nodes looks like that. So it's a value multiplied by a weight, another value multiplied by a weight, and then an output. So in this way, with these recurrent neural networks, we can look at patterns over time. As I said yesterday, this can be a pattern in a value, it can be seasonal variation in a value, but also we can use these for language. Because if we can encode language, we can see what's said at a given time point in a sentence, or a paragraph, or a paper, or a book, or an entire internet, and work out what's going to be said at the next time point. And the longer that time series event goes, and the more detailed the information, the more we approach a large language model. So a large language model takes everything that's been said, uses very complicated time series analysis with lots of inputs, possibly has other functionality in there, long-term, short-term memory, which is something you could look up, but essentially puts those into a deep neural network. So what this is doing is this is encoding a time series set of events. We're encoding things over time. And then once we take that, we then pass that to fully connected layers. So the next layer type is the one that's close to the end of the deep neural network. That's a fully connected layer where we've encoded things. The encoded features are here. And then we feed them through effectively mathematical or pattern detection layers. Each one of these is a hidden node, a lot more complicated than the one I showed you the other day, obviously, but basically it's taking inputs, multiplied by weights, summing those weights, passing them to the next node and doing the same thing. So it's not any more complicated in terms of the concept behind it. It's just repetition of the same thing a lot of times. What we then have is another output layer. There's an output layer that can be single or multiple outputs. The node is usually linearly scaled, but you can use other functions to scale it. And then this is a decoder function, because this is kind of the network's interface with the real world. What is the probability of this happening, or what is happening over these series of multiple states, and are they, changing in different ways? So that's pretty much deep neural networks. There are other functionalities that are more rare, but these are the common ones that are generally available. But whole books and whole courses are run just on deep neural networks. So again, I urge you to have a look at what other functionalities might be encoded. You will not have to write these. Libraries have already been built. You can run some of these in Weka. Look at the help of Weka, look at videos of Weka for deep learning for J. Start to become familiar with these things. Okay? Right. So that's deep learning. The concept I want to move on to now is one of inference. Inference of networks. So everybody here, as I said at the introduction of the meeting, everyone here is made up of thousands of genes encoding for hundreds of thousands of proteins. All of those things are potentially interacting with one another. So that's a very complex thing. The key point to make here is biology is not just a list of features. You are not just a list of proteins, but those proteins are interacting. So when we start to understand the network nature of biology, we can start to understand what the drivers of a system are. What are the switches that tell us, or switch things on, switch things off? You can imagine that biology is not a single note, or a single note in series, but it's like an orchestra. It has lots of different instruments coming in and lots of different things playing, and those work together to produce what is an organism. So that's important to have a concept of that. So what we're trying to do is understand the association between things in a network. So one simple method we can use to do that is we can use clustering. So what clustering does, it gives us the distance between every individual in a population, but it also can give us the distance between variables. Across a population, how similar does this variable look to this variable? How close is the variation in space? And this is a paper which we wrote with the University of Leeds a number of years ago, 2012. And there we were looking at the various estrogen, progesterone, androgen receptors, their interaction with one another in male and female breast cancer. Because we wanted to know what was the hormonal change, how these hormones were differentially interacting between those states. So what we can say is, clustering works on distance. Hierarchical clustering tells us the distance between any pair of things. Yeah? So what we can say is, from the hierarchy, we can say molecule X interacts with molecule Y with a distance A. So we can read that off the hierarchical plot. So, sorry, these are not particularly clear. these are two forms of progesterone. The distance between those is around 35. So that distance tells us how associated that pair of molecules are. Yeah? These two molecules here, these are estrogen receptor beta. So those are very tightly coupled in female breast cancer, but here the distance between them is much greater, or a fair bit greater, in male breast cancer. So by looking at the distance on this dendrogram, we can work out how coupled two proteins are. Yep. So that gives us a simple network. We could draw a diagram in Cytoscape which uses the interaction value as the distance. So that would say, estrogen receptor A with estrogen receptor B has a distance of this, progesterone A with estrogen receptor B has a distance of this, and we could plot that in a wire diagram, dendrogram. Okay? That's the simple form, a simple way of doing it. The problem is there's no location, and it's a distance, not a function. So there's no direction between of interaction. A interacts with B the same amount as B interacts with A. So that just gives us one weight. But biology is more than that. There are feedback mechanisms. Mechanisms shift and become stronger in one direction than the other. So that has its limitations. We can use another method. So we can plot correlation matrices. We can plot X versus Y and get a correlation coefficient. You can get a Y equals mx plus C plot. So molecule X here interacts with molecule Y with a gradient value A. So the gradient, remember, is like the weight of the neural network. That's important to remember. But also we have kind of a measure of how accurate that is. So we have an R value, which is like the error correlation coefficient. So we have an interaction value, which is the gradient, and then a kind of a measure of how certain we are in that, which is the R value. And we can do that across multiple pairs of features, multiple pairs of genes or proteins. Again, this is unidirectional. The correlation coefficient, if we plot something on the x-axis and versus the x-axis versus the y-axis, or if we flip them around, the correlation coefficient will still be the same. So we don't gain any directionality. But there is a function there. We can relate one to the other because they are linear. But it's a linear function. So it won't always be as accurate because biology isn't so linear. The interaction things are non-linear. An extension of this is multivariate linear regression. So in regression we can use multiple x values to predict one y value. So we might get W, if we have 4 variables, W, X, Y, Z. W equals m of x plus m of y, y plus m of z, z plus c. So we're getting a gradient component from each of the variables in terms of their relationship to W. And we get a correlation coefficient. So we have this matrix of functions. In this, we can take the whole thing a bit further. Because now we've got a multivariate model, we can have a directionality on there of our gradients. Because, for example, if we look at xw, here we've got a gradient term for the relationship of x to w, but if we look at x, we've also got a gradient term for the relation of w to x. So we have 2 gradient terms which represent directionality. You have a gradient term going one way, but because it's a multifactorial model, we have a gradient term coming back. So we can show directionality on this. So this approach is the next step of the iteration towards network inference. It has directionality, but it's functional, but it's linear. So it's a linear multivariate regression. So in this context, can you remember what neural networks are? So, as stated earlier on, neural networks are a multivariate. What sort of regression are they? Nonlinear. So neural networks, this is a multivariate linear regression. Neural networks are a multivariate nonlinear regression. So we can start to use neural networks in this capacity and move to network inference. So we can start to explore that further. Another method for network inference is a Bayesian method. So Bayesian theorem is about probabilities of things happening and how those probabilities change when you have other things in there. So if, let's say, the probability of me having coffee with my breakfast is 60%. Unless I'm in India where the chai is better, in which case it becomes 20%. So you see, one state has modified that probability by another thing. So those modified probabilities are changing how likely something will happen. And that's the basis for Bayesian networks. It's how do probabilities change in different states. So we end up with a network of probabilities. So whereas we've looked at the distance as a way of associating things, which is clustering, we've got the weighted correlation coefficient. So we've got the gradient, which is the regression-based method. And now we have the third way of representing a network, which is a probability. And the way, I'm just going to illustrate this, is we have somebody that presents with breathing problems. So that's the center of our network. First of all, we have, has that individual visited Asia? Because the question is this tuberculosis or lung cancer? So the first conditional probability is, does that individual, have they visited to Asia? Therefore, that shifts the probability towards tuberculosis. The other question is, do they smoke? If they haven't visited Asia but they smoke, then it's more likely to be lung cancer. If they haven't been to Asia and they don't smoke, then it's more likely to be bronchitis. So we then, on that, have a probability scale of whether it's lung cancer or tuberculosis. We then have confirmatory post diagnosis of X. So if you get a positive X-ray, that changes the probability. So if you've got an X-ray that shows tubercules in the lung, that's more likely to be tuberculosis. And then if you've got dyspnea, which is like inability to take deep breaths without coughing, then that also shifts conditional probability away from lung cancer towards bronchitis. So you see, having this network of things, we can add effectively probability weights to that, and we can apply these analyses automatically to proteomic data, transcriptomic data. So if this gene is upregulated and this gene is upregulated and this gene is upregulated, then there's a high probability this gene will be downregulated, and so we can start to build those Bayesian networks from that. So the problem with Bayesian approaches, though, is that it's unidirectional because it's based on a probability, and it's non-functional, so it's a probability, not a directly interrogatable function. You can't plug numbers in. All you get out is a probability of something.
So, in some conditions, the probability is 50%. Like, big average.
Yeah.
How does it?
Well, it'd be seen as 50%, so because the weighting will be scored as 50%, it's an inconclusive. But that 50% may be subsequently modified by other things later on down the network. So if everything's 50%, which is pretty unlikely, then you've got a problem. But in reality, things get modulated away from 50-50 because they're changed by other variables as they're added into the network.
So what do we mean by saying that it's non-functional?
So functional means that we are able to extract a mathematical formula from it. We can plug numbers in and get numbers out. It's sort of semi-functional because if you've got a probability of something happening, you can sort of predict. But you can't say, for example, whether it's a sigmoid curve or whether it's a more linear curve. You've just got a probability. You can't predict something directly from it, and you can't interrogate the shapes of the curves in it. So that's why it's non-functional. So what I'm going to show you now is an ANN-based inference method. And this could be easily run with Weka because it's. So if you have a set of inputs, you have an output. You can extract the weights. Remember how I said about parameterization methods, how we could use the weights to parameterize a neural network. And this is what we're doing here. We build a model, we parameterize it, and we have a weights matrix, and then we build our interactions up from that weights matrix. But instead of using genes to predict lung cancer, non-lung cancer, we're using genes to predict genes. So in our first model, let's say we have 20 genes, we're predicting one gene. We train that model to convergence, with methods I've shown you already, extract the weights, and then we can see the change in weights across those 19 inputs that predict the one, and we can start to see where the strong associations are. I'll go into some more detail. So the ANN interaction algorithm predicts the relationship between markers in a subset. We've extracted those using whatever means. Multiple markers are used to predict the expression of one marker. So if we have 20, 19 are used to predict 1. We then turn the dial round, and then another 19 are used to predict the next one. Another 19 are used to predict the next one. I'll show you that diagrammatically in a second. This tells us whether how markers regulate with one another, whether they're bidirectional or unidirectional, whether they're inhibitory or stimulatory. So is there a positive relationship or negative relationship? Is it a strong relationship or a weak relationship? Does it go in this direction or this direction? We can use this graph. So one of the concepts that occurs in drug discovery is what's called a knowledge graph. And that's how we link everything together associated with this disease and with these drugs to work out what the key drug target should be. And this is generating that knowledge graph for us. So I'm going to show you a case study which was used to identify drivers regulating the proliferation system in breast cancer. This approach is multidirectional and functional, and what you can also see is we get these very defined hooks where certain weights, certain interactions are extremely strong, but the vast majority are relatively weak. So we can quickly pick out the key features that we want to include in the network. So, as I said, I think there's 16 here, but how we do this, how we run this. So here we've got 16. We use 15 to predict 1. So it's a neural network. I've not shown the hidden nodes in here because it just gets complicated. But essentially we have inputs and outputs. We then train that model. And here the thickness of the line represents the strength of interaction. So we train that model, we parameterize it. This is a really strong interaction. This one here, you can't see it because it's really weak. Yeah? We've now moved on to our next model, where this is the output, this is used as an input, and all these others are used as inputs. We're just turning the dial round, so the next thing becomes an output. We train that. So you see, by turning the dial round, we're able to build up a matrix of interactions. Everything ultimately gets used to predict everything else. So we get a complete picture of the interaction space. It's based upon multifactorial models, so we can have directionality in there. It's based upon nonlinear models, so we can cope with nonlinearity. We can even slice into the model to look at residuals and see whether some people fit the model for a particular node or don't fit the model. So it's digging deep into the biology. We can colour these lines then we would normally plot these in Cytoscape. This is an interesting story. I had a PhD student who came from Bangalore. And we started plotting such graphs for his PhD, and I could never work out why he was getting different results from me. Well, in warm countries, red is a negative number and blue is a positive number. But in cold countries, red is a positive number and blue is a negative number. So we had this whole colour confusion where he was thinking things were positive, I was thinking they were negative, and everything was just turned the other way around. So we ended up using red and green, but that has problems because some people are red-green colourblind. So we spent quite a bit of his PhD deciding on colour schemes for these networks to view them. But it is only viewing them, so that has to be changed because we actually want to numerically deconstruct the network as well. I'll talk about that in a second. So we can use Cytoscape to change the structure of the nodes, and then we can start putting directionality onto this because we have bidirectional unit and links. So if the weights from this node to this node are really strong, and then the reciprocal weights are really weak, we know that that's a unidirectional influence. If the weights are about the same, then we have feedback loops. So this mechanism will show feedback loops within the network because the weights are about the same. You can have something which is positive this way, negative that way. Negative, negative is common. Positive, positive is more rare. What tends to happen in these networks is we get a lot of negative links because that maintains homeostasis. You know what I mean by homeostasis? Yeah, so it keeps the steady state of biology. You don't want wildly aberrant biological networks cycling and going massively out of control. It's all about keeping that balance. So here are some real results. So this is what a network looks like if we just have 100 features. There are 9,900 interactions. It's a ball of string, but it just starts to show how complicated we are. You know, you've got 20,000 genes which have a map like this. So we filter it to see what's going on. This is applying a level 5 filter. If you stare at this long enough, you'll see leaping dolphins, wonderful sunsets, all sorts of things, and probably go slightly crazy. But it does show that we're starting to see white space in the gaps between the network. If we filter much further, this is a level 25 filter, we start to see patterns. Firstly, it's homeostasis, apart from one node here, which is positive. So all these things are downregulating one another. What we see here also is two hubs. These are things which the system is trying to strongly regulate. The system's trying to shut those up, in this case, to downregulate them. It can be because they're aberrant in disease. Cancer particularly, the system sometimes is trying to shut things down and failing. because there is a mutation to a particular gene, the signal they're trying to shut up. So what we do also is we have a network of interactions, and we want to find out what the strongest interactors are. We can view those in Cytoscape, but it's much better to get a numerical value. So what we do is we take the sum of the values leading to each thing, and that is how much they are influenced by the network. And we take the sum of the values leading from each thing, and that tells us how influential they are. So each molecule can be influential, and each molecule can be influenced in a system. And the most influenced things are the things that we are interested in, because if we start to change those, we start to change biology. They're the switches. They're the thing at the centre of the spider's web, which if we hit, brings about a change to the whole web. What we can also do is we can start to look for connectivity. How connected are these things? How stable are these things? Each one of the nodes in the network has a correlation coefficient of the predictions. Is it tightly correlated? Is it less tightly correlated? Are there subsets of the population that don't follow the rest of the population? They're outliers. In a cancer setting, there might be individuals in the population who are just behaving differently because they have a particular mutation. By doing this, we're quantifying the network and quantifying the individuals in the population in the network. And so from that, we can start to understand what people are doing. How do they behave? So this is not quite so clear, but you can see if we plot the R value, plot the correlation coefficients of different nodes. This is a pretty dysregulated node. Predictions for the model are not very good there. But then this one is tightly regulated. So some nodes are stable across the population. Other nodes are less stable. So that allows us to build up a network, to quantify and interrogate a network in order to find things that are important. The next lecture, I'll go on to show you what's the time? I'll check. Oh, it's okay, we've got time. I'll go on to show you how some of these things have been used. Right, I'm just going to take a 5-minute break, but I'll be back shortly. That's right. Up until recently, AI has been considered a black box. We put numbers in, we get numbers out, we don't know what happens in between. And that's one of the problems of deep learning. We can't easily interrogate it because it's so complicated. So we can't easily see what's going on. So that creates problems. So what we need to do is think about how we can make AI explainable. So explainability is important. As I say, machine learning often criticized as a black box. I saw a paper in 1986 called Opening Up Neural Networks, Shining Light into the Black Box. So that's, this is not a concept that's new. But it's still, I've spent most of my career trying to make black boxes transparent. So data feeds into the algorithm, makes a prediction, but we don't know how it does it. That's a common problem. Oops. And the problem with unexplainable machine learning is we need to get an understanding of a system. Now, biologists are about understanding the mechanisms of biology, how biology works. If we've got a big complicated network which is not transparent, we don't gain any understanding. We don't gain the relationship between cause and effect. We also need to validate the findings of the ANN. So how can we validate that this has a positive influence on this if we don't know that it's a positive influence? Or if we don't know exactly what's happening, there might be other things happening in between. We also, if we are following good practice with making models, we have to understand where their strengths are, where their weaknesses are, where their limitations are, what conditions they work in, what conditions they don't work in. So we have to understand how they're working, what their strengths and weaknesses are. If they're not explainable, we don't get that. So the real rationale behind explainable AI is it builds trust. If we can see how it works, we know how it works, we can build trust. It mitigates risk. There's a big risk of overfitting with deep learning. If we can understand how much of that overfitting occurs, where it occurs, how we can limit it, that mitigates the risks. If we know about how the machine learning is working and is transparent, we can improve performance and we can improve accountability, because ultimately, if we're looking at drug discovery or diagnostic technology, we need to state to the FDA how the system works. We are accountable for understanding the biology. If we've got a black box, then that biology is not understandable. So this is kind of the current state, in my view. Most people, so the research I've done, especially in companies, 98% of companies are using large language models for their biological interrogation of data. They're finding features based upon LLMs. The whole world has jumped onto ChatGPT, but may have jumped from something much better. And if we look at the analogy of how LLMs can discover things from a corpus of literature. So a corpus of literature is, let's say, every medical journal there has been, or every biomedical journal, every plant science journal. So in this state, we have those things that are discovered and are in the literature. So all those published things, we have a body of knowledge, which is, there's a slightly, it's not so clear maybe, but it's a yellow box at the top there. That's what we know. If we apply LLMs to that body of knowledge, we may be able to find new patterns, new interactions, new features, new associations that haven't previously been seen. Nobody can read every single paper and hold all of the knowledge in every single paper in their mind at one time to synthesize a result. An AI can. So taking all that knowledge into account, we have another block here on the iceberg, and this is what can be discoverable by using LLMs to interrogate the literature. Now, there is a problem. The literature, if you have a strong belief in it and it follows good scientific principles, then things are reviewed on their merit, not on their subject. The problem is things don't always get to the public, get into publication. If I write a paper that goes against, say, a particular hypothesis, if I get people who are reviewing that paper who have a contrary hypothesis, even if I present a very good argument, because it undermines their work, they're likely to reject that paper. There is bias, selection bias in the literature. The other problem in the literature is we don't present negative results. I looked at all these studies and I couldn't find any pattern in them. Nobody publishes that paper. The third problem with the literature is we haven't done that study yet. If you think about where we are, all you've seen about proteomic technologies, all you've seen about the advances in molecular profiling, all you've seen about how we can interrogate that data, well, that's only probably 0.01% of what's in the literature. Most of the literature is people doing t-tests on ELISAs. And that's a very reductionist way of looking at the problem. So all of the stuff that's there is pretty old tech. The LLMs look at that and bias what we find towards that. So it's my view, and I'm quite confident of that view, that there is a whole block under the iceberg, which are the de novo discoveries, the new discoveries we can make by doing proteomic, mass spectrometry, expression array, RNA-seq, single-cell RNA-seq work on samples, and then interrogating that data using machine learning methods. That opens up a whole possibility of new things that can be discovered, and they're being discovered based upon evidence in specific data rather than non-specific evidence in the literature. So that's important to have that view of the current state of affairs. One thing after another this morning. Right, that's better, thank you. Just ran the risk of running out of power. Oh, okay, thanks. So let's consider the challenge as well of deep learning and explainability for omics data. So I've said omics data will struggle because the deep learning has lots and lots of things that predict. You know, I've shown that if you take 1,000 random numbers where there should be no pattern, I can predict a random number with about 99% accuracy with a deep learning model. So how generalizable is that? Well, it's random, so not. Shouldn't be able to predict, but I can. That's because of the sheer complexity of the deep learning model, fully connected model. If, however, I constrain that model, I can make the model work so it picks the best things which have the optimal performance. So we kind of have kind of a yin and yang. We have the ability to predict, but if we have a good ability to predict, we get too many things. If we constrain our ability to predict, we get a better separation between the things that predict well and the things that don't predict well, which is what we want ultimately. So we will have problems in generality of models. We have problems with false discovery. We'll find things that are not real. We'll have problems with compute time. You know, deep learning models often require high performance computing, which can be a whole bank of hundreds of machines with hundreds of GPUs. There is a problem with redundancy. If I look at the activation energies in this network, I can take the activation energies across the population for each node, and I can regress them against one another. So a long time ago, we built a deep neural network, and we did that. And what we found was that around 90% of our deep neural network activation energies had a correlation coefficient of 0.95 or more. So basically, they're just repeating the same element again and again and again. And we actually did remove 95% of our hidden nodes, and we maintained a good performance. And the whole process speeded up around 1,000 times. So there's a 1,000-fold increase in speed. We weren't training redundant elements. And the last thing about deep neural networks is if we keep them simple, we can explain what's going on. Explainability means we can get a mathematical formula for our network. We can extract maths. We can publish a formula that shows how these things relate to this question. Other people can try that out. They just plug the numbers into the formula. We will get improved accuracy. So I'll just go over this again. Remember our approach. We have an adaptive system that takes an input, that predicts an output, that compares to what actually happens. That gives us an error. That error is used to update the system, and we keep cycling around and around and around until the network reaches convergence. So the network error drops like that for seen data. That's what we're doing. We're training to get to a lower error. Yep. And we use early stopping to get to that bottom point, and we want to stop because we don't want to get up here where the network's overfitted. By the way, Weka, if you use the Deep Learning 4J module, you can do early stopping in that. I should have mentioned that yesterday. So the goal is to create a model that's not overfitted, that has early stopping, that has the best possible performance for unseen data. Now, I've criticized deep learning, but how do we do it? How do we use machine learning in omics data? Well, our view is that instead of having one massive model that tries to encode, decode everything, our view is that we take lots and lots of tiny models in a swarm. So in a swarm, each model considers a gene on its own and a randomization of the data on its own. And that gives us a partial view of the problem when we solve that bit of the network. We then create another member of the swarm for a different gene with a different randomization. And by creating, in some cases, up to 10,000, 20,000 models in a swarm, we can start to interrogate those and see what's going on. Now, let's talk a little bit about that. So first of all, we can make a swarm. What that swarm does, and this is what I showed you in the latter part of Weka. We were making a model or multiple models on each gene in that list. So by doing that and interrogating the results, which we did in that spreadsheet, we were making a swarm of neural networks. So by doing that, for the whole transcriptome, we get a distribution of errors for unseen data. So this curve is showing how those errors are distributed. So they're ranked, first of all. So each point on this graph is a gene, or even transcription factor, or a non-coding region. And what we're able to do is, let's say we ask a particular question, cancer, non-cancer. We work our way through a data set, testing every single gene with a seen data set and an unseen data set, and plot the performance for a gene on an unseen data set. If we plot that distribution, we get this sharp hook here. So what's in this hook is a subset of genes that are much better at predicting than the rest of the genes. So here, this subset of genes are the things that answer the question. And we do that for multiple repeats, so we end up with multiple curves like this. So we take a scan, if you like, of the gene transcriptome. We randomize, we take another scan of the transcriptome, we randomize, we take another scan of the transcriptome. And we build up this body of knowledge based upon this swarm of neural networks. So this is what that looks like. So we have a data set on tissue or blood or whatever. We randomize it. So we split it into training data, which we use to train the model. We have validation data, which we use to stop the training. And we have test data, which is used to test the model for that particular feature. We then apply that, assess every single gene, rank on performance, and then we get this enriched area here, what's in red. I showed it on the previous graph. So that has a membership of genes that are good at predicting answers to that question for that random view of the data. We then shuffle the data again. This is called Monte Carlo cross-validation because it involves an element of randomness, like shuffling cards or throwing dice. And we redo that process. So we have another enriched list with a different view of the data. We keep doing that. We usually do that up to 20 times. So we randomize 20 times, get a gene rank 20 times over 20 randomizations. And what we then look for is the commonalities in those members. So if the genes occur, what are the genes that occur in that sharp hook that crop up again and again and again? Do they keep coming into that hook? Are they consistently explaining the answer to that question? And what we end up finding is there shouldn't be anything in there, because if you're looking at the top, say 100, 200 out of 20,000 genes, for top 200 out of 20,000, to keep the math simple, you've got a 1 in 1,000 chance of finding a gene in that. If you do 20 repeats and you find the same gene in all 20, the chance of that happening is 1 in 1,000 to the power of 20. So conditional probability is really tiny. It's not going to happen by random chance. Yet regularly we find multiple features coming up again and again and again, just breaking that random chance, which means if it's not random, there must be a pattern to it, which means that biology is important. So it eliminates false discovery. because for something to crop up, it has to crop up again and again and again, and it maybe has to crop up again and again over multiple data sets, because we can do that. We can integrate multiple data sets for the same question. So what does a swarm-based solution do? Well, it trains large numbers of smaller, less deep neural network models. Each model takes a different view of the data and addresses a different part of the problem. We can then integrate that and present all that together, and we can show overlap between the findings, cross-validate them. We use early stopping, regularization, and Monte Carlo cross-validation. So that means we're validating for generality in that whole process. And this is really important. So GPUs have effectively lots of compute units on them. So a GPU can have 1,000 compute units. That's like a little CPU. If you've got a deep model, what it tries to do is spread the whole model over all of those compute units. But the compute units have to talk to each other, which slows the whole process down. With this approach, we run one model on one compute unit, and we do that in parallel 1,000 times. So we can get a massive speed up in terms of how we assess these things by using a swarm spread over a GPU. To the point we can do 50 million molecular combinations an hour. So that's very quick, very fast, because of the optimized use. We consider all of the transcriptome from a single gene product. And then we work our way down and filter out and find the real things that answer the question. So we can assemble the model, we can reduce redundancy, we can increase speed, reduce overfitting, get a better understanding of features, and then through concordance reduce false discovery. So this approach is overcoming a lot of the problems of a deep learning network. So I'm now going to illustrate this swarm approach with a case study. And that case study is looking at common proliferation markers for breast cancer across multiple data sets. So we use 3 data sets in this. We use the Nottingham cohort, and this is quite an old study. So these are using expression arrays, microarrays, or gene microarrays as it's called. So we had a Nottingham cohort with 128 replicates on it. We had an Uppsala cohort of 249, and we had a Metabrick cohort of 1,980. We then validated this separately on a TCGA cohort. So TCGA is a massive repository of proteomic, transcriptomic, genetic cancer information. It was part of the Cancer Moonshot in the US. So this is how we did this. This is the flow diagram, if you like. So we have a data set. We looked at multiple indicators of proliferation. So that's grade, mitotic index, Ki67, survival, TOPO2, MIB, KIF2C. Each one of those we ran as a separate neural network and got a rank order across multiple repeats of each one of those features. We then did the same thing here with the next data set, with the next data set, with the next data set, and each time we looked at just the top 100 and top 200 to see which features were associated with proliferation features. So it's kind of interrogating the data. There's no one true definition of proliferation, but by using multiple definitions and then integrating the results, we kind of get closer to the ground truth. So we did this. These were some more detail about the markers and targets. I'm not going to go into those particularly today. And these are the distribution of those features over the data sets. And this is where we get to the result. So across 3 data sets in the first instance, across 5 or more questions, looking at the top 100 and top 200, looking at the top 100, we found 34 out of the top 100 genes came up again and again and again. If we looked at the top 200, 68 came up again and again and again. So these are consistent features that are there despite the different data sets, despite the different questions, despite the different randomizations. So why did we think this idea up? Because every idea has a seed or origin. Well, what we started to do, we were looking at models of different things, and we kept seeing the same genes coming up again and again and again. So we thought there must be something in this. Let's build this into a workflow to use that to our advantage. So let's have a look at some probabilities. You're finding these common genes in the top 100 across 5 questions, across 3 data sets, by random chance. Well, for a single gene, the probability of finding a single gene in the top 100 in 5 questions across 3 data sets is 10 to the minus 39. And that's calculated by 100 out of 47,000 to the power 5, because there's 5 questions. And that's the first data set. Then 100 out of 22,000 to the power 5 for the second data set, and 100 out of 48,000 to the power 5 for the third data set. So the same things were, this is the probability of finding a single gene in common. We found 34. So that value here can also be increased to the power 34. Now, I did that in Excel because even with a 16-bit machine, no, 64-bit machine, it rounded up to 0. So it's a value with around 100 zero. It's less than 10 to the minus 100. So that's more than there are stars in the visible sky, probably more than there are grains of sand on all the beaches in the world. So that's the chance of finding 1 grain of sand that's purple in every single grain of sand on the whole planet, or 1 grain of sand that is of particular characteristic. There probably are purple sands, but you see what the chance of it being a false discovery are so low. So that we can use to our advantage. Where these 30 or 68 genes, what did they come up in? How did they compare with what other people found? Well, there were 54 genes out of the 68 that are known to be associated with mitotic cell cycle. That's a GO process. 25 with chromosomal segregation, 33 with cell cycle, 27 with microtubule processes, 25 against protein assembly complexes. So those things are coming up. Molecular functions, tubule binding, tubule activity, adenonucleotide binding, and motor activity. So these are all about the mechanics of how a cell divides, how the cells are pulled apart. Obviously, when a cell is dividing quickly, those mechanisms are changed. And proliferation is how quickly cells are dividing. And when cells are dividing too quickly, it's more likely that you get partial separation of the nuclear material, which means that you don't get true copies of the cells, which means you get various genes mutating, and you get what's called chromosomal instability. So a lot of what we find is associated with chromosomal instability. So there are 100 genes in the SYN attractor. We found out of our 68, 53. Genomic grade, SYN 70, PAM. So we get a lot of overlap with what other people have found, but we've also got unique features. And that's important, because if we just found everything as unique in the whole list, then there's no validation there. But if we're finding things that are associated with the question as have been found by other people, that validates the new things we've found as well. So this is that same network which I showed you earlier. This is we then went on to validate this with immunohistochemistry. So these are cytoplasmic stainings with strong and moderate. This is really interesting. Oops. So this is looking at cytoplasmic expression and associated with anthracycline response. So anthracycline is a chemotherapy for breast cancer which attacks highly proliferating cells. So if we know and have an index for predicting how proliferating a set of cells are, we can use that to predict how somebody is going to respond to that chemotherapy. A personalized medicine, if you like. So what we did is we looked at outcome. And this outcome data is retrospective, but we have outcomes for 10 years in this cohort. So we looked at SPAG5 nuclear expression. And what we see in nuclear expression is low expressors tend to do better than high expressors when they're treated with anthracycline. If we look at cytoplasmic expression, then it flips. Low expressors of cytoplasmic expression do much worse than high expressors. So if SPAG5 is basically, if SPAG5 is in the cytoplasm, not the nucleus, which is where it should be, people are more likely to respond to anthracycline. So we can use that, and it's a way of understanding the disease more. And sorry, yeah.
This prediction has this been tested clinically as well, like in ER-negative breast cancer patients, if the cytoplasmic expression was high and anthracycline was low, so has it been tested clinically as well?
Yes. So this is a clinical cohort. And then we went on, you'll see in the paper in a second, we went on to validate this in 15,000 cases in a Lancet paper. So, good question there. So, yeah, we ended up validating this. We went to everybody else with their survival data, with their anthracycline treated cases, sent them a monoclonal antibody so they could stain their tissue microarrays, got the data back, and these were cases across Northern Europe, Canada, USA, New Zealand, Australia. I think there were Southeast Asia, South Asia cohorts in there as well. And then brought that all together, and we could see that actually this predicted, as we say, anthracycline chemotherapy and protein expression as well. So with machine learning, we'd got to this new target of proliferation. In fact, based upon this paper, the Lancet said this is probably a better predictor of proliferation than the current gold standard, which is Ki67. So another way which we can use swarm-based learning is for diagnostics as well. So we can iteratively add to our network. So we pick the best single gene using our methods, and then we add everything else to it as a single gene to find the best pair, and then the best triplet, quadruplet, quintuplet. So we add and build up a panel of markers in an additive fashion. So we can say, what is the best combination of genes that predicts this question? So we pick the best single feature and then sequentially add to it. And again, we have these multiple layers of cross-validation, and we can build this into a piece of code. So what we've done here, this is a graph. We've got the sensitivity, we start off with a single gene. The error sensitivity is about just over 50%. It's pretty bad at predicting on its own. And then sequentially adding to it, we can take that predictive ability up to, if we take 25, up to around 85%. So we find the best combination by sequentially assessing every pair, every triplet, quadruplet, quintuplet, sextuplet, and so on across the whole data set, across the whole transcriptome. And in this way we can start to build panels of diagnostic markers and build decision support tools. So one of the things we've done here, for example, is to look at chronic obstructive pulmonary disorder, look at a panel of markers and predict whether somebody is heading towards an exacerbation. Are they likely to have an episode of COPD? How long is that going to be before that happens? And that's just a piece of work that's just about to come out in the press, in publication. Another place where we've used this, we call it Precise, is in identifying diagnostic biomarkers of latent TB. So by taking whole blood from patients, we were able to do transcriptomic profiling. We had 3 states. We had active TB, latent TB, who were symptom negative, and healthy. So we took the data, and with 4 markers, we had an AUC of 0.95. So we had 98% sensitivity, 88% specificity. So we're able to again optimize a diagnostic panel. Discriminating control versus latent, we could also discriminate control versus active and active versus latent. And then we also modeled, using the network inference, the transition between latent TB and active TB. And we could start to see switches that were coming on when people were moving towards active disease. We then worked and developed this and validated it in a small Chinese cohort, 50 cases. This was with a hospital which people hadn't heard of until recently. It was in Wuhan, in China, Wuhan Pulmonary Hospital. We started our trial in 2019. Halfway through our trial, we had COVID hitting. It was a pulmonary hospital, so needless to say, those people were impacted, and sadly, some of those people in the clinical team died from COVID during the course of that study. So that's quite a sad state from this trial. Nevertheless, the result worked, and hopefully we can bring a diagnostic that helps people in TB. So this is patented, and we're now talking to a large diagnostics company about taking this panel and the algorithm forward as a way of diagnosing large-scale populations for TB. Another area we've applied this is Alzheimer's disease. So in Alzheimer's you have healthy, you have post-mortem confirmed Alzheimer's disease, and you have mild Alzheimer's, which is called mild cognitive impairment. So the healthy really is a misnomer, because this is brain material from dead patients' post-mortem samples. So I don't know, we shouldn't really use the word healthy because they have died. They're not healthy. But they are controls. They were confirmed not to have AD when they died. So what can we do here? The idea is to understand the mechanisms of disease, to build diagnostic methods that help us ultimately go towards blood, where we can build a diagnostic test for blood. But the data was out there. We used it using 5 markers. We ended up with a training performance, so it predicted 94.85% accurately. So it was tissue material. It took us 4 weeks to discover it, and we found a 4-gene panel. Actually, it was a 5-gene panel. And we had sensitivity of 96%, specificity of 94%. What's interesting here is we plot the probability distribution of what is the probability of an individual having Alzheimer's. So we get this big block of individuals up here, which are clear, high probability Alzheimer's cases. And then we get a few individuals just starting to look more like healthy controls. Likewise, all this block here are controls. There's a few controls which are false positives. So they don't have Alzheimer's, or they were not diagnosed with Alzheimer's, but they might be heading towards Alzheimer's. And then we applied the blind mild cognitive impairment cases into the mix, and most of those, with the exception of one here, most of those fell between disease and healthy. So they were more like Alzheimer's, but they weren't as probable as having Alzheimer's. Excuse me. So that's ending. Next lecture. Any questions? So this is how we can use machine learning, omics data, proteomics, transcriptomics, blood, tissue, how we can use this data for diagnostic purposes and build diagnostic decision support tools. So any questions? Yeah.
So you told us about risk of false discovery. So how much it is effective in reducing the false discovery?
So could you speak up a bit? I can't hear you. I've got a fan just here. Do you want some microphone, please?
Sir, you told us about the internominal distribution for the reduction of the risk of false discovery.
Yes.
So how much it is effective in reducing the false discovery, and with respect to the DIA and DDA, how would it be differentiated?
Right, so yeah, it's changing the probability of false discovery by giving us effectively a calculated value. So what is the calculated false discovery rate if we look at a deep learning model? Well, we don't really estimate that, but what we would have to do is compare what we found to a distribution. Or we found 200 genes of import out of the whole transcriptome. That would give us a probability. We then compare that to the probability we get with this new method, and we see a big decrease in the probability. So it's changing the probability of false discovery by quantifying it and allowing us to be very selective. We don't get that quantification in the deep learning model. In terms of DIA and DDA, that's a different false discovery. Has the gene been identified correctly by the mass spectrometry instrument? What we're saying here is the gene or protein, so it's protein, the gene for the DIA, DDA, is this protein or gene, is it really associated with that question? So it's two different terms, same term, but different contexts. So for proteomics, is it a real protein that we've found? Does it exist as a real protein? Here does it exist as something that really predicts an answer to the question? So that's where the difference lies. Okay. Any other questions?
So in your swarm models, if suppose we have different biological study entities, like for location or something like so that different models will be specific to that entities, or it will incorporate all entities and randomly selecting them?
If we've got data for the same individuals, like proteomic, transcriptomic, genetic, well, then we can mine all of those together. So we can allow the genes, the proteins, the transcripts to compete with one another to find their best combination that predicts an answer. So as long as it's in the same data set, it doesn't matter. We can also, if we've got spatial data, we can tag location as well as MZ value, or location as well as gene identity, together, and then mine that as a spatial question. So we collapse the whole spatial data into a single 1D list, but by tagging the names of the location onto the names of the genes.
Suppose like it's in single unanswered data we are using it.
Yeah.
So like other meta information, we'll just tag it along.
Yeah, we can tag that on, so it might be an important feature. What I would suggest is probably tagging it on and then not tagging it on and see how the results vary. Because if you've got whole transcriptomic data, then in theory that all together should explain the answer to the question. If, however, you've got other variables that are explaining the answer to the question.
Yeah, suppose like other RNAs, microRNAs.
Yeah, we can include, well, we do, when we do a deeper analysis, we'll include all the non-coding, all the microRNAs, all the transcription factors anyway.
And what is the probability if we get a small set of data as compared to the general AI size data? Like in small cohorts or something like that?
Yeah, so small cohorts, because we have an independent framework for assessing the probability of false discovery, what we tend to see is if we've got lower numbers, then it becomes a bit more variable. Now, the pool of things that are in that stable region drops because we don't have a full or better, such good representation of the system. Whereas if we've got around a couple of hundred individuals, then we get really good stability results. Out of the top 500, the best I've seen is 460 features. Out of the top 500, consistently predicting the answer to the question across 20 different runs of the model. So that's pretty stable. Sometimes we might not find stable things, and we've got lower numbers. It might be as low as 20 or 30, so we have to bear that in mind. But we can, with this, compare the quality of results from multiple data sets as well. So say we get 3 data sets. First data set we get 250 features, second data set we get 180, third data set we get 300. By looking at each one of those feature lists, we can say, well, data set 1 and 2 really overlap strongly with one another. They are in the same space. Data set 3 is very different, so it's possibly an outlier data set.
How will it sound if we do it for different entities separately?
Yeah, if for different entities,
Separately and then merge the all in a network. Like
yeah, we could. Well, if we don't have the same values for the same individuals, we can run that separately, but then look for commonalities in the gene name. So the gene name, protein name will be the same. So let's say we look at proteomic data, transcriptomic data. In this one, it's proteomic data. We find 200 genes, it's 200 transcripts. In this one, we find 200 proteins, and we find 60%, 70% overlap between those. We can see where those features are stable between proteomic data and transcriptomic data. Because it's data-based, all the theory behind how the data is generated, as long as we normalize correctly, isn't important, because we're using the information content of the features, not how much they are variable, how big the differences are, but how much information that's relevant is there in each one of the features. And that's quite a shift away from the way things are conventionally done. But it has, as we've seen, has yielded very useful results. So. Okay, any other questions?
So exactly on what basis do we choose which cohorts we have to use?
Purely numbers. So if you've got 5 data sets, which have got over 200 individuals, use them all and see what is common between those 5 data sets. Because the more you look at these, the more data you look at, the more and cross-validations you do, the more robust the things you will find are. And that's that cross-validation that's really important. If you consider sort of 6,000 individuals across 5 cohorts, compared to 100 individuals over 2 cohorts, that result is going to be much stronger for the bigger number. So, yeah. Okay. Any other questions? Is there anything anybody wants me to clarify, go over again? Can you see the logic behind what we're trying to do here, how that's working? Yeah. Okay, great. So, I'm going to go into the next lecture now. Well, we may have to stop for lunch, but we'll carry on after lunch if we need to. So previously I talked about understanding, in a diagnostic sense more or less, the features of a system, or the features that drive a system. So we effectively asked the question, could we use this to discover biology that might be a useful thing to develop a drug target for? So the one we selected was P53. And we selected P53 as a target because firstly it's commonly of tumours. So people who have a P53 mutation in their tumour are less likely to do so well. It has a role all those things. It's a bit like a Swiss Army knife of a pathway in a cancer setting. So this is the P50 With this approach, we modelled the p53 pathway, we worked out the drivers of the p53 pathway, and then we looked at how druggable those drivers were, and if there were other molecules which we might be able to utilise to target them in the pharmacopoeia, the body of knowledge around pharmaceutical entities. So the question here ultimately is, can we develop an in silico pipeline for drug discovery that leverage the power of omics data, big data and machine learning? In other words, can we digitize drug discovery? So again, this relies heavily on this distribution curve, which helps us with false discovery issues, and we can use this method to enrich by concordance and stability. Except now, rather than using it to answer a question around phenotype, so rather than say using it to predict healthy versus lung cancer, we're now using it to predict a gene in a pathway. So each one of those, each one of these green boxes now becomes an output for a neural network. So I'll show how we do that in a little bit more detail. Okay. So rather than showing the whole P53, what's going on here? Never mind, we'll see. Rather than showing the whole P53 pathway, which would look really complicated, I'm just creating a simplified network to show how we do this. So let's say we have a hypothetical pathway where molecule X is known to link to Y, which is linked to Z. Molecule X links to B, which links to C. Molecule X links to molecule A. So that's a simple pathway. Each one of these blue dots is a gene, and they're thought to link together in a molecular pathway. So what we can do then is we can then start to mine the interactors for each one of these members of the pathway using this approach of looking at distribution curves. And we can mine the whole transcriptome to see what things strongly predict that target in that condition. So we can start doing this mining for a breast cancer data set. So we find the interactors of molecule A or X. We then do the same thing. So it's going a bit slow at the minute. I have a whirly wheel on the screen. I don't know why I'm going to go slow. So we then do the same thing for the next molecule. The next molecule. So effectively, sorry, it's on going very slowly. So effectively, we map the interactome for every node in the whole pathway. So you're gaining evidence about how things are interacting using the known molecules of the pathway as a seed to build a network up. What we then do is enrich using this distribution curve. So we now have a set of stable interactors for each gene in the pathway. So what we then do, sorry, this is kind of animating itself because it's going so slowly, is we then look for commonalities between those stable interactomes. So a red dot is something that's found in the interactome of this one and this one, the stable interactome. So effectively, with this new red dot here is a new gene that is an intermediary in the pathway based upon... Oh, sorry. Computer's going wild. I don't know what's happening here. Right, I'm just going to use this way to present. It's less slow. So, as I say, we've got a stable interactome for each member of the pathway. We look for commonalities, and effectively we're gap filling the p53 pathway in breast cancer, so it's highly specific, and but by searching across the whole transcriptome. So in this way, we are effectively gaining new knowledge about that pathway and what might be involved in it with it. If you look at other methods, and if you remember I talked to you about Gene Mania, Metacore, Ingenuity, those pathways are generic. They kind of look at all of biology. You might be able to get a bit of information around certain conditions, but they're generic. They kind of give the vanilla flavor, general biology, whereas this builds the network specifically from omics data, specifically for a subset of cases. So that's useful because it means we can quantify and add to the network. So what we did here is we used this Metabrick data that I mentioned before, and we looked at a clinically annotated primary invasive breast cancer set, specifically focusing on the triple negative. So you may have heard of a triple negative breast cancer. It's ER, PR, HER2 negative. Some histologically call it a basal phenotype. So that was the data set, and we looked at TCGA. And we specifically selected triple negative cases. So when we mined the p53 pathway, everything, let me just get this the right way around, everything in green here, everything in green here is a new feature of that pathway that's come up based upon evidence from the mining of the transcriptomic data. So these are new things. The pathway is more complicated than we realized, and using this method we can add features that are specific to cancer into that system. So what we then did is used inference modeling, and I've already talked about this. But the same process is applied. And what we get to, like we did before, but now slightly differently, is an augmented p53 pathway. So we can see this is a tissue necrotizing factor, this is EGFR. We're getting strong hubs again, that like what we found before. So these are things that are highly influenced in the p53 system in breast cancer. But as I said before, we can do a driver analysis and work out how much influence and how influenced each feature is. So this identifies the most influential molecules. We've done previous studies where we've shown that the most influential molecules, if we change them, manipulate them, we can change phenotype. I'll show some examples from plant sciences in a bit where we created transgenic plants by switching certain transcription factors on and off. So that gave us an indication that these drivers and hubs are interesting things and potentially are interesting for drug targets, as drug targets. So the Lancet paper, we showed those methods there, and we found actually in the Lancet study, when we created a knockout, I'll show this in a second, of the SPAG5, we actually slowed proliferation right down and we changed the centromere structure of the cells. So again, phenotypic changes that resulted from that target. So, as I said before, driver analysis takes the sum to each gene and tells us the sum of genes leading to a gene and tells us how influenced the gene is, and then takes the sum of the genes links leading from the gene to tell us how influential it is. And what's that? This is the one, yeah. The order of this one, I think the top most influential features, there's a disconnect between the name and the gene, but this is the correct value. So STEAP3, CD82, TP73, those were the most influential. These are novel. So these top three are known in the pathway. Just ignore this bit. These two are novel, and we're finding highly influential novel features coming up. If we look at the most influenced features, the names are correct here. So what's interesting here, if we look at the most influenced features, we have our number 2 feature, which is known, and that's a TP53 apoptosis inducing protein. So we've gone through the whole transcriptome, looked at everything, looked at the network inference, and we find features that make biological sense, being highly influenced and highly influential in the system we're exploring. So that gives us some confidence that we're starting to unravel some of the biology here with this method. Now, we can do the connectivity and stability. I've already talked to you about that. Once we have that, we can start to interrogate the databases that are out there for things that might be interesting in terms of their druggability. What we can also do, though, and this is important, is because we make the model and do the network inference on a specific type of data, so triple negative breast cancer cases, we can also repeat that process for healthy breast, for healthy lung, for healthy kidney, liver, skin, blood. And we can basically build differential models of the disease state, from the disease state of the pathway in different tissues. And that's moving towards something called a digital twin, where we can build effectively a model of different parts of an individual and see how the pathway works and how it changes between those individuals, or between the, not between the individuals, between the parts of an individual. And that's important for drug discovery because firstly, we don't want to drug biology that's essential for healthy life. If we start knocking out essential systems, then people have serious side effects and die from the drug. But if we knock out things in the cancer that are not present in the healthy state, then we can start to find useful things that are differential from healthy. Finding, if you like, the molecular causality of the disease. So causality is what we're looking for here. What's the molecule that causes this problem? Or what are the molecules that cause this problem? So if once we have a target, we can drop the target into DGIDB, the Drug Genome Interaction Database. That uses a combination of curation, so curated data sets and curated databases, and data have been mined from DrugBank, from PharmGKB, from Chembol, and from the Drug Target Commons. So these are all sources of drugging information for molecules. So genes are here categorized as potentially druggable, as based upon their membership in pathways, functions, families, ontologies, the Human Protein Atlas, and the indicative druggable genome. So this is found at DGIGB. So rather than showing you some results, I'm going to use this as an example. So let's pick a strong driver, GAD45B. So if I go to Chrome, I go to search DGIDB. This is the Drug Genome Interaction Database. So let's pick some things from this list. So let's say GAD45B, that's a starting one. And then let's pick another one, TP53 ALP1. And I'll pick a third. I could put up to 50 in here if I wanted to, or more than 50. Let's do TP7, integrin beta 1, ITGB1. I'll pick that one. ITGB1. Yep. And I'll search. So it's picked up 3 genes, it's found them. There's 4 potential interactions that are inhibitors. There are 3 potential interactions that are antibodies. And this is our table of results. So what I'm interested here is in interaction score. So I'll sort by interaction score, largest to smallest. So first one, integrin beta 1. So this is volociximab. Sorry, you have to get your tongue around databases as a drug name sometimes. So volociximab is an anti-neoplastic agent. So that's quite good because neoplasms are cancer. So this is good for treating cancers. Integrin beta-1 is a target. It's not been approved for that target, but it has a pretty strong interaction score. So I could take, let's say I want to find triple negative breast cancer, I could suggest, given the role of this molecule being strongly influenced in breast cancer, let's try a trial where we look at volociximab and its treatment for breast cancer. I can see more about volociximab, so let's see what I can get from that. So volociximab, well, mAb at the end of a drug means monoclonal antibody. So the volociximab is a monoclonal antibody targeting this biology. It's got a Kemble identity. It's been used up to phase 3, but there's not any FDA data linked. There are other interactions with integrin alpha-5 as well. So that's a potential target. Let's see what else we've got. So we've got, these are unique matches. GAD doesn't seem to be any good. It's not particularly druggable. We've got a few monoclonals. Is that one? Oh yeah, it needs to be resorted. We've got Prayagraj. It's an anti-inflammatory agent. It's got an interaction score of 2.91, which is not too bad. That could be another potential therapeutic target, knocking out ITGB1. So we've got some interesting things going in here. When the scores start dropping, so these are less good, we probably wouldn't look at those. If 4 isn't bad, but we can see sometimes scores up to 20, 25, 30, and those are really strong. But often they tend to be the drug that's specifically used for that molecule as a treatment. So they're already an occupied space in terms of the IP. So we can use DGIDB to look at that. Sorry.
Can that be used to study other type of interactions like drug-drug interaction or drug-protein interaction?
That might be better solved by Kemble, because Kemble would be drug-protein and drug-drug. This is about, well, it's protein-drug interactions because it's the protein product, gene product, which is the protein. So you can use that for gene-protein, so drug-protein interactions. But for drug-drug interactions, it's probably better to look at Kemble because that has the chemistry part of it. So, yeah. If it's drug interactions via molecular or protein gene intermediary, then that would do it. But if it's direct drug-drug interactions chemically, then you're better off with Kemble. So that's DGIDB. What we can also do is we can apply these things to druggability indices as well. So DGIDB is one example, but there's also a piece of software called DrugGnome AI, or Drug Genome AI. So Drug Genome AI is from AstraZeneca, and it's a way of working out how druggable is a small molecule. So if we look at that, so it's, if we do a search on drug gnome AI, it's served through GitHub, which is the developer approach, developer way of sharing things. Okay, how are we doing? Yep, that's good. So it's loading. Taking a bit of time. So if I click on the home, there we go. So I can search for a gene. So if I put ITGB1, oops. You can see it comes up with ITGB1. Right, so now it's interrogating this database and it has various scores. So it puts these into different models, and these are whether they have reached different phases of clinical trials. So has there been, for this target, a molecule that's been taken to a particular phase? So has it been taken to the clinical trial phase, the chemistry phase, and then you have different tiers. These tiers are defined here. So T-clin, let's see if it wakes up, is approved, clinical trial approved drugs. Yep. So my T-clin score is 97. So something has been taken to clinical trial for this molecule. T-chem, small molecule activities have been shown in Kemble. There's approved drug targets with chemistry. Tier 1 is targets approved for biotherapeutic use, and Tier 2 genes that encode targets with known bioactive drug-like properties. So you see, as you go up the tiers, so they're more likely to be clinically approved drugs. But this is scoring pretty well with high probabilities across all those criteria. So that's making this look very suitable as a potential drug target. We also get where there are potential interesting areas of method for treating. So we've got small molecules that scores 97.8%, a raw score 99.99% for antibodies. So it's clearly antibodies, we know that, we saw those earlier, and for PROTACs. So a PROTAC is a drug modality which is drug antibody conjugates. So where you bind a drug onto an antibody, direct that at a cancer, and when the antibody hits the cancer, binds to it, it releases the drug. So it's quite encouraging for this target in terms of that. We have various scores for small molecule antibody and small molecule in the cancer and non-cancer space. So CPD, clinical phase small molecule drug candidates in oncology. So we know we're targeting breast cancer, so that's a potentially interesting offering. And then this is showing the modalities where it might well work. It shows the cellular location, so there's intracellular and membrane, so that's why it's a good antibody. And there are orthologs for this in other species, so if we need to create an animal model, we know there are orthologs there. And we can see here where it's expressed. So we can estimate where other effects may occur if we start to mess with this. And this is the expression of the protein in different parts of the system. Oops. So if I don't think drug GNOME AI actually gives that. Let's have a look. Can I right click on that? No, I can't open that. But I can look at that in maybe Kemble or DGIDB, because DGIDB is doing that function primarily. Yeah. The real place you could look for that is open targets. So if you want to know the drugs that have been approved for that molecule, then you can search that in open targets. So, sorry, I'm jumping around. So drug GNOME AI is based on Mantis machine learning. Looks at disease-specific gene druggability framework. It uses a semi-supervised learning approach based on Keras and TensorFlow, so it's a Python or R-based package ultimately. I think if you go onto the website you can download the package for it. And then the publication is Communications Biology 2022. So it's publication if you want to look that up. This is what I was saying about the digital twin concept. So this is visualizing it. So we have a network. That network is quantified. That network is being built for a particular data set. So we can take our P53 plus network, which we found in cancer, and then by taking those same genes into network inference in, say, healthy breast, we can build a quantified model of it in healthy breast. So now we can put the cancer pathway side by side with the healthy pathway and start to see where the differences lie. What are the differential drivers? Ideally, we want something that's a strong driver in cancer and a weak driver in healthy, or a strong receiver in cancer and a weak receiver in healthy. So that starts to help us whittle down the key targets. But what we can also do, and we've gone out, and it feels a bit like the body snatchers, but I'm doing it digitally. I've gone out and collected all sorts of healthy organ data from publicly available databases. And then I can use that to apply these network inference models in different states to build effectively a pathway model in different healthy tissues. Because basically I want to be able to drug something that's just in cancer, just in this case just in breast cancer. But I don't want it to have an effect on colorectal tissue or effect on blood or an effect on the brain. So by building these digital twin models across different tissues, I can then rank my molecules to see which ones are important in the disease, but not in all these healthy states. Nearly done. So we can use AI for molecular docking as well. So we can look at 3D structures, and we can look at drug molecules as structures. We can also, so two ways of looking at drug efficacy. One way is to look at the structure, and if you remember yesterday I talked about 3D convolutional neural networks being used for docking. Another thing I can do, and I've seen companies do this, is every molecule is represented by what's called a SMILES code. So that's a string of numbers, letters, which represents the unique structure of that. Now I can mine that SMILES code, if I put it in a framework, to see which elements of the SMILES code indicate something that might bind to my molecule from the literature. And then I can use that SMILES code in a model in a generative way to find new structures, new SMILES codes that might lead to molecules that could be created that haven't yet been created. So by looking at either the 3D structure or the SMILES code, I can start to build molecular docking models. Traditionally, we would get that protein structure from X-ray crystallography. So basically, we take a protein into a crystal form in a resin or a matrix. We fire X-rays at it, and based upon the shadows that are produced by the interference patterns of that protein, we can start to work out what the structure might be. Because different parts of the protein diffract X-rays in different ways. So alpha helixes diffract differently from beta pleated sheets. So by rastering across the image, across the resin bulk, changing the orientation, rastering across that, we can start to build up a 3D structure. That's the traditional way of doing it. But more recently, of course, we can get structures from AlphaFold and other pieces of software. AlphaFold isn't the only answer. Just because they won the Nobel Prize doesn't mean that the research in that area completely stops. In fact, there have been 5 or 6 other iterations of AlphaFold that have come out since that. Hence the word "and similar." So we could model SMILES codes with LLM and foundational AI to build some binding predictions. So that's pretty much how we can utilize this process, going from data, proteomics, transcriptomics, through to enrichment. So what are the molecules? What is the biology that associates with this question? Through to pathways. What's the biology that associates with this pathway? Through to inference. What are the most influential, most influenced molecules in this system? Through to druggability, through to drug design and drug discovery. So by doing all that workflow, we can create an in silico pipeline to find new targets at the cost of electricity, because there's that much data available out there already. But don't stop producing data, people who are using mass spectrometers, and don't stop producing transcriptomic data, because we are literally a millionth of the amount of data that really would fully represent the human system. But we can use what we've got. So in this way, the idea is we can get to new treatments for diseases much more quickly. And we can get to new drugs much more quickly. Because the conventional pipeline, as was, it would probably take us around 15 to 20 years to find a molecule that might be a drug target. And all the validation, all the knockout studies, all the studies of association through a disease model, because invariably they weren't real human tissues, they were cell line models or animal models. All of that would take around 15 to 20 years and cost a huge amount of money. Then, once we've got the biological target, it would be another 15 years to probably take it to clinical trial. So that's a pipeline that takes 30 years to get to benefit for humans. The technology is moving so quickly, that pipeline could be out of date within 6 months. There's also a 95% failure rate and a multi-billion dollar cost to get a drug to market. So it's on that landscape that we're starting to do these things, because we think there is going to be a much better way of doing things using machine learning. And that's the basis for how we do things. So, this is my NTU email address. I have an ARU email address, but I'll just leave that there if you want to contact me. But I'm going to be carrying on teaching you. Are there any questions? Yeah. Yeah, you may have to speak up because this fan is really to consciousness. Yep.
So basically, what Professor Penrose has told that regarding quantum gravity.
Quantum gravity, yeah.
Basically, what he told that consciousness is a thing which originates from microorganisms that can also be synthesized from the protein, but that is a debatable. Actually, consciousness is still a mystery. There are many theories. So I want to know what's your thought.
So consciousness, by its definition, is probably quite continuous. In the words of Einstein, though, how can we define consciousness when we are within that consciousness ourselves? Because we can't solve the problem of consciousness because we are within that consciousness. Although we can contemplate what a consciousness might be, but you know, if you feel familiar with the principle of Tao in Chinese, as soon as we seek to define that which is Tao, then we cease to be able to define it. And could we argue consciousness is a bit like that, in that we can get so far in defining consciousness, but then by trying to define it, then we cease to be able to define it. It's a philosophical question, but I think we can replicate elements of consciousness. We can get so far towards defining it.
What I'm trying to say is like AI is getting advanced, so is it possible to solve it by quantum computer physics already? World Deep Mind is working on that.
So that's a good point. So at the minute we really have memory, but do we have consciousness? And we probably don't have consciousness because these things are incapable of self-awareness. So intelligence, or an aspect of intelligence and consciousness, is self-awareness. Is a computer, just because it's good at mapping and modeling language, aware of itself? Probably not. There has to be, and this is where it gets to be a bit of science fiction, because we have this AGC, Advanced Generative Computing, AGI, sorry, Advanced Generative Intelligence. Where does the spark come in an extremely complicated computer for it to develop from memory to consciousness? That's a kind of a quantum leap. I mean, what is consciousness? And consciousness can be defined by animals, by anything at a certain level. You said about even at the protein level.
There is no measurement of consciousness.
Excuse me, sorry to interject. I think we're going a bit way more. And we have some other time. We are chasing few sessions, and today we will be finishing off our sessions at 4. So those who have registered, please proceed with the lunch. And while you're at lunch, you can discuss if you have problems, whatever you want to discuss with Professor Graham.
conceive what would be the, what would you start to build?
You have a set of overexpressed genes.
Yeah, maybe 1000 and 700 are inhibited or underexpressed or whatever we use. So there is a fewer. So what would you start building if you were to solve this problem? You are in the year 1972. What would you start building? In order to make sense of the data. So you will use a database. So you have to build a database first, right? So we always we rely on databases. The entire omics workflows, any omics, transcriptomics, proteomics, metabolomics, anything that has omics to it would definitely have a database associated with it that tells what is what. So for MaxQuant, again we had a database. We usually take the proteome database, and then based on the strips in digestion patterns and mass by charge ratios, we match the theoretical and the practical values, and we tell this is probably a protein. We are never very sure, because there is always, even at 1% FDR or 2% FDR, we are not 100% foolproof, right? So that's why we go for validations. But suppose you want to understand, there are a few things we do, and one of the stuff that will be studying today is reactome. And what does the slide say? It's a database of human biological pathways, right? So those who are working on algae, you have other databases. There is David, there is other databases, but it's usually important that the wider your model system is used, the more easy it is to do omics. If you are starting off with something extremely new where nobody has worked, most probably you have to start the old school way, where you knock out a gene and try to see what function is doing what. But if you're using a widely used adapted model, so this reactome PowerPoint is not mine, first of all. It's taken from the reactome database. They have a CC license, so already they have a. So they start with this paper that, suppose you're studying about a pathway, and they talk about multiple proteins, so caspase 8, CD98, procaspase 8, and you see they tell a lot about the pathway. Now if you're having multiple proteins that are interacting with each other, you have multiple methods by which they're going up or they're going down, you want to take a systems approach. You want to see how overall this gene expression going up or down, or sometimes when we do not know, we have a very few DEPs, we want to understand how this dysregulation is impacting the biology, right? And for that we have databases that already have the pathways mapped onto it. So we know B comes after A, C comes after B. So this way we have the pathway mapped. Reactome is one such database, and it is actually very beautiful. So Reactome is a free, online, open source, curated database of pathways and reactions in human biology, and they are authored by experts biologists. So you don't have to they don't rely on stuff like ChatGPT, because they hallucinate. And anyway, it was conceived much before LLMs came into the picture, and you have an extreme detail. So all the reactions are rightly mapped till the cellular compartment where this reaction might happen. So suppose you have a specific protein that is located in the nucleus, and in the mass spec data you're saying that it has gone up. Reactome can tell you it's a nuclear protein, and it might have these probable interactors. So it will take into account all these factors, where the protein is, which pathway the protein is a part of, where this pathway usually occurs. Is it a cytosol? Does it occur in the cytosol? Does it occur in the nuclear? Is it a membrane protein involved in signal transduction? And what it also does, it gives you beautiful pictures about it. So it is extensively cross-referenced. Cross-reference, I think you understand. So it takes up information from multiple databases. It's UniProt, KB, PubMed, Ensemble, the gene ontology, Entrust Gene. So multiple databases, they take all these data, try to build a common tool where all these data sets are brought together, right? So, and since it is extensively cross-referenced, a good advantage of a cross-reference database is that it does not have much room for error. So what it can do pathway analysis, which will be our focus today. It can do expression overlay, it can do species comparison, and also it can use to infer orthologous events in 17 other species. So we started off telling that this is a human, this focuses primarily on humans. But suppose you have rat data, right? But you don't do much of your studies on in vivo studies. You will use either a mice or a rat or maybe a zebrafish. So in that case, the differentially expressed proteome would not be from humans. Can you use reactome? Of course you can. But then you have to use the option where you will map the component, the gene component to the, there is an option. When we go to the actual tutorial where you will use data sets, then we can see that how we will use a rat data set. We will try to map it to orthologous events in 17 other species. Of course, rat is one of the species. So, in theory, pathway steps are the units of reactome. So there are multiple pathways. You see, there can be a binding event, there can be a degradation event, there can be a transport event, a phosphorylation, dephosphorylation event, or some other classical biochemical pathway. So these are all in reactome. When we talk of units of reactome or events, these are separate events that reactome maps. So this is a reaction 1, that is example of an enzymatic pathway. This is another example of transport of calcium from platelet dense tubular systems in the cytoplasm. So you can see here, you can actually see the other reaction. This is a binding reaction, this is a dimerization reaction, this is a phosphorylation event. So you can actually map all these events onto reactome, that these are the events that are happening, and it will take your gene expression data set or protein expression data set. So you can work, reactome is a tool that will work for both transcriptomics as well as proteomics data sets. And how does it take evidence? It takes both direct evidence or indirect evidence. Direct evidence, of course, since it's a human-centric pathway database, direct evidence is anything that has been reported in a study on humans. But most of the studies are never, most of the times studies are not, you don't get studies directly on humans. First you would get evidence from maybe a model system. So it can be a cow, it can be a mouse, it can be a rat, it can be a monkey. So those are indirect evidences. And what other source that does it take? It uses gene ontology, both molecular function, compartment and biological processes as one of the source. KB, so UniProt, Ensemble, PubMed. So these are the external sources from which reactome takes data, and we had discussed this. So they cross-refer across multiple, so they will go to the gene ontology database, look up on a protein. Then it will also look up about the same protein on PubMed and see what are the literature events that are available and how well do they correlate. If they correlate well, you will get on your report that these are the reports. And the reports are actually that we get from Reactome are actually very advanced and very detailed. So how much is the availability? So almost 52.6% of the 20,296 predicted human protein coding genes they have predicted. So, but still, I think this number is quite an old number. These numbers. So every Reactome version, if you go to the version notes, you can actually see what is the curation level for that version, and it keeps on increasing, especially with the advent of AI. They can do that. And how do they do the curation process? So they have experts who are recruited, reviewers again who are recruited, and they both report to the staff who maintain the database. So it's humanly created, humanly maintained. So pathways are reviewed by expert biologists. Curator works with the author to represent the knowledge in the Reactome data structure, and there are always regular pathway updates. So when we will open Reactome today, very soon, I can show it on the PowerPoint on the projection screen. Later on, you can go back home and you can try with the data set that we will share with you. So when you go you will actually, if you go to into the details, you click on those small I and small stars, you will see what is the current level of curation, who has curated. So everything is properly documented, and every 3 months they release a new data set. Or one fun thing, many of you make diagrams, right? Flowcharts. And Reactome also has a very good library of icons that you can use free of charge. So many of you might be using BioRender. BioRender is paid. Reactome is free. But since it's free, it has its own limitations. It does not have the vastness of BioRender, but for any pathway schematics that you need to do, you would get everything, right from the cell to the mitochondria to the nucleus, transmembrane receptors and stuff. So, as I said, it can project onto species. So, suppose this is the original reaction that they have found. In mouse, they have found a similar reaction. So it will be projected. Now, A is not present in Drosophila. Since it is not present, there is no ortholog, the protein is not projected, the reaction will not be projected. And it will show up on your data set that there is no projection of data, right? It will show up that these proteins could not be mapped. And what are the species? Right now you have Homo sapiens, Arabidopsis, Bos taurus, Caenorhabditis, Canis, Danio, Dictyostelium, Drosophila, Rattus, Plasmodium. You have Oryza sativa. So those working on plants, you can look into whether you can project your data can be from Arabidopsis or so mostly it's animals, but there are two important plants. That is, one is rice, the other is Arabidopsis. Okay, they also do some disease annotation. So it can show infection, it can talk about cancer, it can also talk about metabolic diseases. So these are the larger classifications. So infections like it can be HIV, influenza, botulism. Then you have cancer, you have different types of cancer based on the signaling pathways. You have metabolic diseases. So this is a influenza virus life cycle that you would find how Reactome schematically represents it. So point is, when you would see in your own data, this is something that you would totally learn by doing. When it comes to your own data, if this particular protein is expressed, this box will fill up. And it will start showing that you have evidence at this level or you have evidence at this level. Right? So at this point, it's just a blank template. The moment it starts analyzing your data, once you start clicking on this individual pathway, you generate the pathway report, it will show you what are the pathways that have been mapped onto it. Again, this is for a cancer. This is the level of details that you get in the pathways. It will immediately take your data, project onto the pathways. So when you're trying to build a hypothesis from discovery data, Reactome is a good place where you can start, both by making sense of your data, second, by building a good, strong hypothesis for a targeted experiment. Now you want to block a signaling pathway, you want to do some siRNA stuff or some CRISPR-Cas stuff. So you know where to do and where to start. Right. So again, you have disease display for phenylketonuria. So this is here, it will start displaying that this is where things are wrong. So if I go to the Reactome tools, primarily we can do overrepresentation analysis in pathway topology and expression overlay. We can also do species comparison, molecular interaction, and interactive pathway. But primarily this time we will be doing overrepresentation. And what is overrepresentation? Anyone? Mathematics? What would you say overrepresentation? So what is the gender ratio in India? Around 50-50, like. So suppose you have a class full of girls and there is one boy. Can I say that girls are overrepresented? For that, of course, for that particular class. Right? So when you try to understand any pathway, suppose for a specific pathway there are 100 proteins involved, right? And in your data set, out of all the differential expressed protein, you have 200 differential expressed protein, 90 of them are mapped onto that specific pathway. So usually for one pathway, for these 20,300 genes, out of 20,300 genes, there are 100 genes responsible for one particular event, one pathway, right? Even if you take like a baseline value of 20,000, out of 20,000, 100 genes are responsible for one pathway. And then out of your differentially expressed genes or proteins, suddenly you find out of those 300, 90 of them are mapping onto a specific pathway. Can you say that this pathway is overrepresented in your data set? Because what is the probability? By random chance, just by random chance, if you were to pick things up, right? To make your calculation easier, let's make it that for there is a species where they have 100 genes in total, right? And one event takes place, it requires 10 genes. So by random chance, there's a 10% probability, out of 10 times, you would pick up a gene that is responsible for event A, right? In your differentially expressed genome, you have 6 genes. And out of those 6 genes, 5 are genes related to event A. So does it fall in the random chance category? It's not by random chance that only those specific genes are repeatedly coming. So what should be 1 in 10 has now become what? Like almost more than 60%, right? You have 6, or in your data set, you have 6 genes, 5 are now mapping onto one that. So that is overrepresentation analysis. And when we talk of differentially expressed genome or differentially expressed proteome, this overrepresentation analysis gives us a very strong idea about what has probably gone wrong in our model system or in our disease system. So this is the pathway, this is the website. All of you can note it down. It's reactome.org. And this will be your total pathway browser. So when you open this reactome.org, you will land your landing page. This is your landing page. You have this hierarchy. Here you can select your species. You can search for the diagram. This is the pathway panel. This is the analyzed data, video tour, layout. So this is primarily the landing page of the pathway browser. And when you do your analysis, you get a pathway overview, right? And you see, this is how the pathway is shown up. They have shared pathways of the edges, and then it shows exactly where this pathway is leading to. So this is where things start getting interesting. You start getting biological insights from your data. And in the hierarchy, you can click on the specific diseases and pathways that you want to focus on that has been overrepresented. So these are the diagrams. So you have positive regulation and negative regulation. You can get into this contextual informational panel. You can click on it. So the point of it is it's a tutorial, right? So I will not waste much of your time. You already have shared in the morning. This is the same thing that I've shared in the morning. So you can go through it. And so most of you would not be working on human data sets. So there you can do a species comparison and you can map your data sets, whether you have an ortholog for that specific protein. You can click on the protein's numbers to get interaction. So if you are familiar with any social media site, it's much easier than the social media clicks. Till now I'm very confused. Every day they keep on changing the user interface. So if you can handle that, Reactome is much easier. You don't need to know coding. All you need to have is a set of data. I think if the internet is working, which it is not. Reshma, I need a login password. It's working, right? When you are trying to analyze proteomics data, we go to analysis tools. So what it does is merges pathway information, the overrepresentation data. It also maps your data. If you're not using data sets from humans, it will map if there is an event, similar event happening. Mostly people working on rats, mice, Drosophila, Daniel, zebrafish. I think they can use this tool to map. Right. So what we usually do is, this is reaction version 90. So this keeps on changing, and you see you can use any of these data sets. So we have tissue-specific expression, cancer gene data, metabolomics data, microarray data, small molecules, gene name list, unique product accession list. Right? So if I click on it, this will take me to an example data set. What have I put? I have put the list of genes, long list. And if I click, you can click or so project to humans. So all, this is species A, this is human. It will be using this as a background. So every species has its own set of identifiers, which this software usually identifies because of the accession number that we use. We can also use include interactors. So it basically uses the molecular interaction database, and it creates. So suppose you want to do a much more broader analysis, we can expand the analysis background. So even if I click on this, include interactors, and then all I have to do is sit back and relax, and while it will start analyzing my data. So if you go here, you see this is an over representation analysis. And what does it say? It has found 17 identifiers out of a total of 101 that is already there in the database. Right? No interactions found. And when you click on any of these, first of all, it tells you that this is the cycle that is getting impacted, and this is a Creative Commons license, so you can use these tools to create figures for your publications as well. Of course, you have to cite them, but there is no charge as such to use their tool. And best part is you are generating this from your data, so that makes the If I click on analysis, you see there is also an option of not found. So these entities were not found. As I told, they have not mapped all the proteins yet. So you might get some data points that won't be mapped. You need not worry, because if you have 150, 147 have been mapped and you have lost out on 3, very unlikely that you will lose much information. And still, if you're worried that you're losing out on information, there are other tools like Metascape that you can use. You can go to downloads, and you can read on this download analysis report in PDF format. And this report that you get is pretty exhaustive. I'm just waiting for the report to get downloaded. So here you see they have shown multiple pathways. The moment I click on that pathway, it will immediately zoom in to those pathways. So this is extracellular matrix organization. Zoom in further. So this is integrin cell surface interaction. So there are other pathways that are not getting impacted, It's only this particular pathway that has been impacted, at least per the data that you have submitted to the portal. So immediately what were basically numbers and alphanumeric codes starts making biological sense. And which is very important if you're chasing a publication, you just cannot tell that, okay, I had a very costly instrument and I injected my samples. Those samples gave me, I identified 8,000 proteins, and there were other guy had another sample, we identified 9,000 proteins. And yes, since I have identified so many proteins, I would love to publish. No, publications don't work that way. You have to tell them a story, right? So you have to start with the problem and then at least offer a solution. If not a complete solution, even if it is a bad, even if you offer them a story, okay, we tried these solutions, these don't work. Even then that is a good. Okay, one for the other scientists it becomes, okay, these guys have tried, they have failed, let's not do it. So we won't do it. So, I don't know if it has been downloaded. So when you generate so this is from the sample data, I have not used our data yet. So that will give you a very detailed report. Important thing when you are doing your analysis. Again, this is UniProt version 90, Reactome version 90. I think some time back I was working on version 80 something. Versions keep on changing. Version 90 will not give you the same result as version 91 or 89. So when we generate, whenever you use these tools, especially because they are web server-based tools, do your complete analysis, download whatever you can, keep it on a pen drive, keep it on your Google Drive, send an email to 5 friends whom you trust, send an email to your supervisor. He might delete it because he might have 1,000 emails piled up, but at least you will have it on your sent email, right? So keep multiple backups because perhaps you have made a hypothesis based on version 90 and while then you proceeded with all your experiments. Now you have lost the figures. Now you are going 2 years later, every 3 months they release a new version, right? So after 2 years, there are already 6 versions more. Now version 96, suddenly you find your data doesn't make that sense anymore. It's something else. Not always true, but sometimes it does happen that what was your top hit, whatever pathway you had got in the first chance, 6 months later, or 1 month, or 1 year later down in the downline, something has changed. So now your peer reviewer won't believe you. It's always down these reports, it's very important. And what does this report contain? It contains a nice table of contents, which are again, you can click. So you click on introduction. It will tell you about the Henning. This guy is Henning Hammerjakob. He has one of the lead authors. His group who makes. So it will tell you about reactome. Used to come or do this analysis, and here it says 167 out of 172 identifiers, and each identifier is what? Name of a gene or a protein, were found in reactome, and there were 1705 pathways that were hit by at least one of them. So primarily what it tells you that these 167 proteins or genes are components of 1705 pathways. All non-human identifiers have been converted to the human equivalent, and again it gives you a unique ID, and this is valid for 7 days. You don't take the risk of 7 days because life moves fast, and then it shows you this genome-wide overview that which are the pathways. So it might not be visible to you. Let's zoom in. These are high resolution images, so anyway you can You can see muscle contraction, not much hits. Circadian clock, not many. Immune system,
On.
is back in. Yes, a few. Then it comes signal transduction, you see it lights up. Just like this fireworks. And it is actually a fireworks representation. Then you have disease, yes. Hemostasis, yes. Neuronal system, no. Metabolism of proteins, no. Sensory perception, no. ECM, extracellular matrix organization, yes. Cellular response to stimuli, yes. Autophagy, no. Reproduction, no. Cell cycle, yes, part of it. DNA replication, does it impact? No. Metabolism of RNA, no. So you get a genome-wide overview that what are the pathways that your differentially expressed proteome is getting impacted. And suddenly, your data, which was just numbers, you get numbers from the mass spec, you get again some alphanumeric codes in UniProt IDs. Suddenly you now it starts making sense to you that okay, these are the things that might have gone wrong in our data, in our patient. So these are the 25 most relevant pathways sorted by p-value. So constitutive signaling by PI3K in cancer, then the PI3K AKT signaling pathway in cancer. So this was a GBM data sets. What is GBM? Professor Graham has lots of work in GBM. So it's an assignment for you. What is GBM in common parlance? It's a cancer. Right. So it's a glioblastoma. And you see, immediately you have lots of signaling. So EGFR and phosphorylation and extracellular domains. So, so these pathways are sorted by its p-value, and which basically means the higher it is up, in very layman's term, the more higher it's up on the table, the more impact it has on the disease pathway. At least they also have a false discovery rate, which is of course low, and then it starts showing you the constitutive signal. As I told you, once you start using your own data, you can actually start zooming in and see even within that pathway, where are my proteins? So can you see something that has lit up? So let's try again. I think it's already visible, right? So all the pathways, these are the specific pathways that are getting impacted. So this is primarily so each pathway would have multiple protein partners. This analysis can tell you of those protein partners, which proteins are in your own data, right? So a pathway might have 100 proteins, but your input file will have maybe 6 of them, and they will map and highlight those 6. So this immediately tells you which pathway your data set, you can target in your hypothesis building phase. So you have used discovery phase data and analyzed it perfectly to build upon a very strong hypothesis. Can someone call Manmeet among the... And not only does it tell you, suppose you are new to this field it will tell you what does this pathway do. So it will, since it's a completely humanly curated database, it will tell you. So the signaling by this PI3 AKT is frequently constitutively activated in cancer via gain of function mutation in one of these two PI3K subunits. And the gain of function mutation does this, blah, 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 along with all the papers that they have cited. Now, this is not hallucination by ChatGPT, that it will suddenly give you some fake references and look-alike papers. This is strong data set.
मम्मी तभी गए सर कैंपस सर अभी सर पोस्टर के लिए गए सर पोस्टर निकल नहीं पा रहा है सर
कौन सा
सर जो दोनों पोस्टर जो सर डील से ठोक दिया गया है वो सर निकल नहीं पा रहा है
निकल जाएगा टेक हेल्प फ्रॉम CFT गोविंद
मुझसे सर बोला है सर वही बोल रहे हैं
who created it, who authored it, who reviewed it, and also which are the proteins. So 13 submitted entries were found in this pathway, mapping to 17 reactome entities. I think we already discussed what is a reactome entity. Then you move on to the next, and you again see it shows, it is, I don't know if it is visible, but if you can look at the cursor, so these are the proteins that have been hit, and this gives you the p-value. And this is part of reactome disease dataset. Similarly, same. So of all the 25 that top hits that you generate, you will get detailed references, detailed information about what was the protein that was upregulated, I mean at least in your upregulated or downregulated data set. Okay, it will tell you what was the protein doing, which pathway was it a part of, was this a part of a disease pathway, which all the entities it has been mapped to. So this gives you a solid foundation to start your hypothesis building, right? So now suppose you are not satisfied with this, you want more. So how many of you understand what is a meta-analysis? Anyone who does meta-analysis? You know what is a meta-analysis? Anyone? Research scholars. What is a meta-analysis? Kind of, but not exactly.
Collating all the other data.
Collating multiple data sets or multiple tools and trying to make sense of it together, right? So you have suppose for any disease, for any cancer, you have proteomic data sets from 5 different cohorts. Now these are 5 experimental groups. Somebody has done it in Sweden, somebody has done it in India, somebody has done it in Sri Lanka, somebody has done it in the US. Now you get all their data sets and now you analyze them together, and then you try to find what are the commonalities, right? So there are multiple gene ontology tools. Reactome is one of them. So suppose, Shambhu, where is the folder? This one? So this is the upregulated one or the downregulated version? You don't have the FC values. Anyone you have with the fold change values? But before he does that, I will copy a part of it. Suppose this is a rat data, and I want to see what has gone wrong in my rat model, and I want to understand, so I'm not taking any sample data set now, I want to use multiple tools to analyze my data in one go. I don't want to go to Reactome separately, and Reactome is not the end all. Tools, you have Panther, David, and you want to do an analysis in one go. So this metascape.org does the same kind of overrepresentation analysis, but what this database does is, what this software does it takes your data and they will ping all these databases separately, and then they will run their own algorithm when these databases, so Reactome sends them their results, A, B, C, other groups, they send them the results, and when all these results are brought together, they will do another analysis and create their own results. So let's see what they do. So I am just pasting a list of differentially expressed protein, and I will try to do submit. So input species, of course they have identified by the numbers that this is from rat. You want to analyze it as the same species, or you want to project it to Homo sapiens. So I will not project it and just click on express analysis. So while the analysis, it takes some time. So you see, there's lots of papers till date. In 2024, November 15, there have been over 7,000 studies that have used this tool. Also, it has some chat GPT functions, so you can ask any question which you feel that you do not know, but so it's kind of more automated than Reactome. So always use it. The ChatGPT information should always be verified once. Don't use it as such. Also, they have lots of blogs that their team makes. So one is Data Scientist's Notes on Machine Learning. And these are the release notes. So this is the current version, and if you see, it was launched in 2015, and slowly how they have, the current version is version 3.5. Again with them, this is a Chinese, again with them is all these databases, any web server based tool has this tendency to get update themselves. Of course, they cannot be the same version repeatedly. Problem with these tools is you cannot go back to the old version, right? Due to server space, they remove the old version, they completely shift to the new edition. So, and you cannot also download. So whenever you generate data, download all possible figures and images that you can generate. So if I go back to the Reactome result, if I go to downloads, you can see you can download it, the pathway analysis as an Excel file. You can also have this detailed analysis report. You can also have this JSON file, or for all other resources, you can also download the identifier mappings in a CSV format. So if you're planning to use this for your own research, download everything. Keep it safe, give it a numbered file, so that, and in your lab notebook, please write it down that I conducted these analysis. These were the brief results. Maybe you can just take a printout of and stick it on your notebook. And you can also tell that I have send this email from this ID to this ID and this time. Because thing is, 3 years down the line, when you start writing your thesis, in all probability you would have forgotten. And since these are numbers, I myself faced a lot of it, although I was not working on this. So I had a list of primers and I got confused because since I was using a set of cloning experiments, so there were many primers, like a primer, then a nested primer, then the sequences, I got confused. So it took me around 2-3 weeks just to sort out which was the primer sequence that I actually used, because I had ordered 3. Technically all 3 would have worked, but only 1 had worked. So keep a note of whatever you're doing. So I guess we have the Metascape result ready, almost. So once the Metascape So this is what it does. So it gives you a detailed report. So this is the gene list. This is the different types of, since it's a meta-analysis, you see you will have multiple gene ontologies. So gene ontology, biological process, then you have a reactome gene set, since it's a rat. You have a network of enriched terms. So these are the networks that shows you have a protein-protein interaction data. And since it's, I think you can actually glean more data, the data set was small, and you can again download it as all-in-one zip file, because they have even a shorter duration. So within 3 days, they will delete all your data sets. What you can do is, and same is true for MetaBurnalist as well. Even if you have to use it for publication, those heat maps, you want to use it for publication, they would require a 300 DPI. So go click on generate 300 DPI, download and save it. Don't think that you will do it later, because later you might not even need it. So you see, everything has been downloaded in this zip file. They also create analysis report, and they are even better. They will make you a PPT, which you can directly use as for your lab seminar. So, right. So these were the pathways that were enriched, and what you cannot see right here is they have also given in the notes section that how they have done it. So they tell we have first identified all statistically enriched terms, and can be gene ontology or KEGG terms or canonical pathways, hallmark gene sets, etc., based on the default choices. Accumulated hypergeometric p-values and enrichment factors were calculated and used for filtering remaining significant terms, but then hierarchically clustered into a tree based on the kappa statistical similarities among the gene membership, similar to what is used in NCI David site. So this is how they have made a list of the pathways. Can anyone tell me what could possibly be the tissue source? It's quite evident. You have so you see, you have gliogenesis, you have astrocyte differentiation. More often than not, you would find that these things are, you have vesicle mediated transport, you have modulation of chemical synaptic transmission. So in all probability, this was a brain tissue. So you can do forensics as well. Suppose you find a tissue that is too mangled, you can run a mass spec and then do a reactome analysis of the proteins expressed, and then you can. So I will try to share a few more data sets. I think Shambhu, you're here. Do we have that data, that upregulated and downregulated? Can you share? Can you pass on this pen drive to him? विशाल का डेटा है तो दे दो या तुम says that I can give it to you. So it's you all have to do is play around with these tools. So once you get a hang of it, you will start using it in a much better way. And the good part with big data is, since the list of differentially expressed protein here was a bit less, the more proteins you give, the more pathways it maps onto, the better, the clearer the picture becomes. Also, since you have already learned how to use MetaboAnalyst, one thing you can easily do is try to separate the upregulated and downregulated proteins. That will tell you which pathways were probably upregulated, and which pathways are in all probability downregulated. But again, since these are discovery data sets, I'm telling you, always use it to build your hypothesis. Right? Much of it maybe it's not true, especially when you're working with something that is a bit of a heterogeneous tissue like brain. Brain is a bit difficult tissue to work with because there are so many types of cell. There are so many types of neurons. Then there is astrocyte. Then there are microglia cells. So they are a bit difficult. But if you're working with something like a liver tissue, that is more or less a very homogeneous tissue. Liver is homogeneous. Lungs is mostly homogeneous. Kidney might have 2 layers, but again, kind of homogeneous. So, but basically, right now, here you have to learn how to use the tool, go back to your own lab, use your own data, and try to see if you can make sense. Anyone who is using proteomics in their research, you are using. So, what's your animal model? Rat. So maybe, what's your genal, right? So yeah, you can use it if you have any differentially, but your problem was you are not getting the protein on the mass spec. So yeah, once you get a signal, so right, so here you have this output where you have it arranged in a log to fold change. So all you need to do is system. using reactome. So this is where I can do this projection that if I need to project into some other species. But anyway, it works fine for rats. And these are proteins. These are uniproteides. So you can also upload it as a CSV file. You can also upload it multiple lists. Don't do that. FASTA will be too big. So you just need the accession numbers. A simple copy paste does the trick. I'm a big zero in metabolomics. Yeah, basically it's for these are for analyzing gene set enrichment, so you can use genes as well. Risha, Shambhu, what is this? This is hippocampus data, right? It's hippocampus. Yeah, part of the brain. So again, you can find that whatever this data was, these were the pathways that were upregulated, or these were the pathways that got most impacted. So synapse organization, chemical synaptic transmission across chemical synapses. It also shows these are the most probable protein-protein interaction enrichment. So for each given gene set, so Professor Graham was talking about string. So there are multiple databases like string, BioGrid, Omnipath, InWebIM. So these interactions, this tool can use all these tools separately and give you a meta-analyzed version of it. So this is how they have created. So they have made figures for you. It's available in these three formats. You can go to this Cytoscape web version. So you can get it as a PDF as well. Ideally it's a Cytoscape session, right? So if you have the Cytoscape software, which I think this does not have it, you can use it, download it as a Cytoscape file and start working on it, right? So you can download it as a Cytoscape file. These are the protein-protein interaction network. So these are the pathways that they have been enriched: synapse organization, modulation, chemical transcription. And all you need to do is just click on.
I'll show the last few slides.
Right, you have showed it? Yeah. So this is all about the tool, and since this is primarily a tutorial, I would ask Shambhu to share the data set with you, and all of you can start playing with the data. Okay? These are tools best learned when you start yourself using it, and these are actually huge tool sets, data sets. I mean, these tools they have lots of features. You might need some, you might not need some. But the best way to know whether you need it or do not need it, like in Reactome, there is something known as the foam view. I have not shut it down. I don't know what was that. Did I shut down Reactome? Right. So you can. So there are multiple types of visualizations. You can open the pathway diagram. You can use this Voronoi visualization, which let go then. So yeah. So this immediately tells you of all these pathways, anything that is highlighted yellow is affected, and the strength of yellow. These colors again you can change. So. This color is also editable, so there are ways, if you go to the settings, you can edit that color as well. And what happens when you click on any one of these is you can zoom in, and we can again zoom in. So, as I said, there is no end to it. You can left click to zoom in and hold for details, and if you again hold it back, it will take you to this figure that what is going wrong, right? So these are extremely interactive. I don't know how to zoom into this. So for this specific factor, now this signaling, you see there is a small yellow. So this signal is not impacted. The signaling by this molecule is impacted, and the level of impact is also shown. So if it is full yellow and dark, that means it's highly impacted, it is half full. So there are multiple ways of interpreting or making sense of biological data. I think that was what the essential theme of this tutorial was. That Metascape, Reactome, even string. I think string is one of those. You can take the same data set onto string and you can do similar analysis. हेलो 10-15 मिनट में आता हूँ. So you can do a similar analysis on string as well, but once you use Metascape, so I think a few of you might know how to use string. So yeah, so you can So again, this is also a database that has lots of levels of curation. So this is like from curated database. These are from experimentally determined. These are predicted interactions. These are other interactions that you can have. Text mining, by co-expression study, by protein homology, and you can click on any of these hubs. So if you can click on it, it will tell you what this protein is and what this data might mean to you. Of course, since it is a big data, I mean it's a large number of data, it depends on what kind of biology you have. You have to have a pre-existing knowledge about what your disease system is, what is known in that and what is not known, and how your discovery data is actually aiding in understanding a new level of understanding of the same disease. Can you have a new, if you have a new pathway that can be a target for any therapeutic intervention. So all these things you can do. I will request Shambhu to share it on the data set on the group, and you have around 30 minutes or so to work on it. Use any tool of your choice and try to work on it, because the moment I leave, you're most probably going to forget. So I think they can have some time till 3:15. We can work on it, and later on Professor Graham will also add on to something, and once he is done, so please take down the notes, try to work on it, and then maybe tomorrow when we have this final discussion or Q&A session, you can have questions. I'm not giving any assignments as such, but yeah.
Can we delve more into a particular pathway to get to know more about the particular pathway? For example, if I'm going into lipid synthesis, then I want to know that which proteins or drugs are involved in that.
So one thing is that you can know more about the pathway, of course, but what I'm trying to tell you is, with this kind of analysis, this enrichment analysis, this overrepresentation analysis especially, it tells you in your data which are the pathways that most likely got impacted, right? And Reactome and Metascape, they will give you these scores based on their confidence levels, that okay, with how much the higher the score, the more these p-values, the higher it is, the better. I mean, it will tell you that okay, there's a fair chance that this did not happen by accidental, and these are the pathways that might have got impacted, right? So if you really want to know whether this pathway is really responsible, think of some pharmacological inhibition strategies, or some CRISPR-Cas based strategies, or siRNA based strategies. Try to see what happens if you block that pathway. If that pathway is hyperactivated, can you use an siRNA or any other method by which you can silence the pathway and you have restoration of the phenotype. So all this stuff you can do.
Usually we help a metabolite sugar how this to be after that. Like if the metabolites are getting reduced, it is inhibitors or not, and that.
I have to take this call.
So just one point I would make as well. I've shown you a set of tools.
Hello.
Those tools side by side. I showed you about switching the pathway, for example, from egg. But you could get that pathway from reactor, or you could look at the phenotypic state.
Oh, okay, sir.
So disease versus healthy. Find a set of stable enriched markers that explain the disease state. Put that into reactor, see what pathway is involved, and then use the pathway mining approach to mine what is going on around those pathways. Because remember, the pathway miner is filling the gaps in the pathway based upon evidence. So it expands that space that you're getting from Reactome or from K and takes it to an evidential level for a specific state. What essentially we're doing is we're moving you from scientists to becoming engineers, because engineers have a whole set of tools available to them to extract, to make, to define things.
And giving you those tools is moving you from scientists into data engineers. So you're able to sort of use a lot of different things to get as much out of your data as possible, to go back to being scientists when you then have a hypothesis that you can take forward.
So here you're building a very informed hypothesis, right? You're taking your own data set and trying to build a hypothesis that is derived from your own. I mean, Shambhu has completed his thesis and he has done some wonderful work where we started off with discovery data, and then he found out that these are these pathways that can be a good intervention strategy for the disease model he was working on, and then he used some pharmacological inhibitors and he got really good results. So it's not that I have seen from somebody else or read in somebody else's paper. I have seen Shambhu work. From 2017 he has been slogging. By 2024 he has some robust data sets and he can tell you that how important it is to have a discovery data. Try to have a discovery data whenever it is possible, because it removes the bias. Right? It's an unbiased approach. When you take the entire proteome or entire transcriptome, you are removing the bias that, okay, I want to look into this pathway because we are very prone to bias. Right? We are humans, we are prone to bias and we will get bias. So if you tell that, okay, I think this particular pathway might be of importance, sure enough, you would find at least 70 or 80 manuscripts that will tell that this pathway is very important.
Yeah.
But is it important in your particular case? You don't know. But then you start projecting what Reactome does. It projects, right? It takes rat data and projects it onto humans. So you might do the same as well. So try to, if possible, whenever you're planning an experiment, if you have the money, you have an opportunity, build upon the discovery data. Sometimes there are freely available data sets, like you can download it from the Pride server or the Proteome Exchange server, to run your own analysis, and then you can build your own hypothesis again. So that's the way. I mean, we are just providing you the means and ways of how to make sense of what started off as small peptides in a small tube, then they became numbers, then they became alphanumeric codes, and now we see we are doing this bottoms up approach where we are breaking things first, the flavas that we have broken, and now we are using evidence to glue them back.
And if you've got your own private data and then you look for somebody else who's done a similar experiment on Pride or Protein Exchange or even GIOD or TCGA, TCGA has proteomic data, GIOD has proteomic data as well, then you can bring your data set side by side with another team's data set and start to look for commonalities between them. If they found it and you found it, then that validates your data, it validates their data. Don't be afraid to think they might be wrong. But then you found the same features. Two separate groups finding the same thing is much more powerful than one group finding something on their own.
So maybe, Shambhu, have you shared it on the group? No. So those who have an access to a laptop, you already have the Wi-Fi password. You can take out a few more.
Everything's falling apart. Everything I touch, don't come near me, I'm dangerous. Okay, so I'm going to just go through some more tools that would be helpful for you. And so the first bit I'm going to cover is, and I've got about an hour to cover this, so we'll probably be finishing at 4:30, so you can carry on with this assignment after that. The first thing I'm going to show you is sources of publicly available data. So, how are we doing? Right. The first one is very specific, and that's specific to cancer. And that's the Cancer Genome Atlas. So, what I can do is here I can click on Access TCGA data. So if you just Google TCGA cancer, it will bring it up. This is part of a cancer moonshot program, and it's all sorts of, mainly American organizations, putting cancer data onto a common data portal. So if I click here on Access TCGA data, okay, there we go. So what they have is they have a whole range of different cancers, and they have a bar chart here which indicates the tissues in which those cancers are involved. So we've got bone marrow and blood, so that's leukemias, various hematological malignancies. We've got bone, bladder, bile duct, brain, breast, lung. So we have a lot of data here which we can access. For each one of these, they have tissue slices with slide images, they have DNA data, they have RNA data, often they'll have proteomic data, and methylation data in some cases. But basically it's a huge repository with large numbers of cases. So say I want to look at lung cancer. If I click on lung cancer, that's created an analysis cohort for me. Lung has been created. I can save that and go back to that at a later date. So it's now said, right, well here are the there are 5,331 lung cancer cases in the total data set. So I can get transcriptomic data potentially from all of those. So what I can now do is if I go to a cohort builder, I can see the experimentation which has done these. I can see the types of cancer here. I can see the organ of origin, the primary site of the tumour, our case IDs. I can look at the demographics as well. So I can select male, females, different ethnicities. I can look at age at diagnosis. I can look at whether they're alive or not, or banded ages. So I can be very specific about what I select from this cohort. I have the general diagnosis. This has stage, grade, morphology, a whole lot of additional things. I can look at the disease status, disease-specific classifications. This one's not particularly fully filled yet. I think they're always updating it. Treatment, exposure, specimen, biospecimen carts, available data. So I'm going to look at available data. Let's have a look there. Is there anything I can use there? No. So I'm going to keep it quite general in the first instance. And then if I click on this, which talks about additional details, I now get to see what's going on here. I get to see what they're made up of, so I can see adenocarcinomas, squamous cell neoplasms, epithelial neoplasms, so different subtypes of cancer. Let's go back on that one. If I go to repositories, I can select different experimental strategies here. So I want to select RNA-seq. So what this now has done is just looked at all of those data sets and come up with gene expression as the target. So for gene expression, I can see that there are 1,871 files, 1,520 cases. In some cases they will have gene expression data for both healthy tissue, well, that is or cancer tissue, and adjacent healthy tissue as well. So once I have these, maybe I want to do something with these data. So I can next click on tab separated values. That gives me a files table. I can see what all the file names are. That's useful for later. But where are we? cart's empty. So in order to use these, I have to add them all to a cart. So the internet's a bit slow. Okay, RNA-seq. So to add all of these files, I don't want all of those files. Fusion, RNA fusion, okay. Sequencing, transcriptome profiling, I want that. Gene expression quantification, I want that. And star counts. So if I go up to the top here, just remove all files from cart. Now if I click add all files to cart, it will add all of those TSVs. So TSV is a tab separated value file. Each one of those tab separated value files will have the expression profile with full non-normalized count, FPKM, FPKM-UQ and TPM. So these are different expression normalizations. I mostly use TPM values, but it will basically give me that for the whole transcriptome, including long non-coding regions and full detail of the expression. So once I have the cases in the cart, I can now say download cart. I'm not going to do it because it will take a long time, but essentially each one of those tab separated values gives me a transcriptomic profile. So it's a way of getting very big data sets very quickly. Just another couple of things. Where it says download associated data, I need to download the biospecimen cart. Because when it downloads the TSV for the individual file, it has a massive long file name. And it's difficult to go from that into a clinical data set. So the biospecimen data file effectively translates the file name to an individual sample name. So I need to convert and add those two together by sorting. So once I've got that, the other thing I have to do, and not many people realize this, but different projects all come together under TCGA, and different projects sort their gene files in different ways. So if I assume I can just lump all these together and then assume that they're all sorted in the same way by the gene identifier, that is not the case. So a lot of people have criticized some of these public data sets because they say, oh, it's a load of rubbish, I can't find any patterns in it. Well, the reason they can't find any patterns in is because they haven't all aligned every single file individually to the genome. If you do that, it starts to work, but if you don't, you've just got rubbish. So that's an important thing to know. Not many people really, well, some people realize it, but some people don't. So that's one source of data. Another source of data is gene expression omnibus. So this allows me to search through a very large amount of data. It's called the gene expression omnibus, but it has metabolomic, proteomic, transcriptomic, copy number variation, methylation, a whole load of different data types. So, and whereas TCGA is very specific to cancer, this is general, it has a whole range of species in it. So anybody who's published data, often they will load it up to GEOD. So I need to search for something. Any suggestions? I need to search for so I've looked at dilated cardiomyopathy, but if you give me something to search for, I'll see what data there is available for it. Any suggestions? Unless you've got some suggestions, I'll put in glioma. Sorry? Sorry? I've got this going on, sorry. Pardon.
So BRCA1, BRCA1.
BRCA1. Well, we're not looking at genes, we're looking at indications, so diseases or... Yeah. Tuberculosis, right. So there are 21,340 studies that have some mention of tuberculosis in their abstract. So needless to say, that's pretty comprehensive. I can sort here by the numbers, low to high. So what I can first do is say this is the most, this is the highest number of things. What you tend to find is the high ones have a particular chip. So for example, this is saying if you look over all of HU133 plus 2 arrays, there's 175,000 replicates. We're not interested in those, we just want something a bit more specific. So if we start to scroll down, then there's some more general. So if you look here, this is SNP, this is a genome-wide association study of tuberculosis on the Chinese population. So there's 1,008 samples there. Blood RNA signature for predicting treatment outcome in tuberculosis. There are 914 samples there. That's RNA-seq data. So someone has taken blood, looked at response to therapy. That's RNA-seq. So I want to see about that. That looks interesting. So if I click on that, it takes me to a summary page. Here there's 914 samples, multiple 100 cases, 38 controls, so they've got multiple time points, which is useful. The contributors are Zach, Thompson and Shankar. So if I want to look at this data, so it's Illumina HiSeq, so it's a pretty good sequencing machine. I click on download RNA-seq counts. I can now get various files like the series matrix that tells me all the clinical information I need to know about the individuals. I've got the RNA-seq raw counts matrix. I've got the normalized counts matrix, and I can get the TPM or the FPKM values from that. The nice thing about GEOD is the data you get is in one contiguous block, whereas with TCGA you have to download the individual files, merge them and assemble them. With this, you just download the block, you download the sample data relationship file, just merge them together, you've got the data that's good to go. So that's GEOD. One thing to note about GEOD is if I look here, I've got the study type. So I've got expression profiling by array, genome tiling, high throughput sequencing, I've got methylation data, I've got protein profiling by mass spec, protein profiling by protein array. So I have got proteomic data in there. Now, if you don't find something on PRIDE or Proteome Exchange, it's worth looking on TCGA, not TCGA, GEOD. So I can select these and show them. I think if there's not many of something, it just shows everything still, but we'll have a look. This bit doesn't seem to work as well as I'd hope it to. Hope for it to. Yeah, so this still shows the same things. So that's GEOD. Another data source which is worth looking at is Array Express, and that's from the EBI. And if I put in here you can see I can look at tuberculosis, and then I can look at extra terms, but I can keep it more general to begin with. So if I look in here at tuberculosis, and now there are 513 results. So human macrophage identifies tuberculosis susceptibility, intestinal tuberculosis, lymph nodes, active and latent tuberculosis. So you see there's a whole load of data sets there. There are human, bovine, mouse and zebrafish studies here as well. I can look at the technologies. So here we've got array assay and sequencing assay, so it's not got proteomic data. The reason for that is EBI also hosts Array Express. Sorry, as well as Array Express, also hosts Pride. So it's just putting them into two separate repositories, and they don't cross-talk in that case. So I can download data from this source as well. It used to be that EBI had an agreement with NCBI, and they would harmonize their two data sets. But now they don't. They stopped doing that about 5 years ago. So you can get different data sets on Array Express that are not necessarily on GEOD. The other one, I don't tend to use Proteome Exchange. I haven't used it before, but we've also got Pride, Proteomics, and again it's the same sort of format. We can put in here, for example, so there are 216 data sets. I don't like the interface with Pride so much because you can't immediately see replicate numbers or technology. But if I click on more, okay. Yeah, so this one, there were 12 items in here. It's just raw spectrum. There's no SDRF. So Pride, in my view, is still got some development to do. I've only started using it recently because it's been quite sparse in terms of data compared to other sources. So that's repositories. I need to leave that one open. and looking at cell models of a COVID system and understanding what the drivers are of that COVID system. So this is what I showed you, the network inference mapping earlier. This is what we get out from that. We get a source, interaction, target value. So we get a gene, which is a source, an interactor, an interaction value, and a target gene. We repeat that, and we then do driver analysis on it, things like that. If we take the average, use that. For cytoscape, what we do is we take the average and then sort it. So if I insert a graph, so this is a typical distribution here in this system of the interaction values. So again, we get some sharp hooks at the extreme. And what we would do with this normally for cytoscape is we can't present all of that because it's just a complete ball of string, you can't see anything. So we would take the top 200 of those, maybe, and we would put those into cytoscape. So what I'm going to do now is I'll just take those, put them into a file. And I'm just going to call this test, and I'll put it into GIAN workshop. Right, so now what I'm going to do is close this session down, and then I'm now going to, oops, so I just want to create an instance of Cytoscape. Cytoscape is freely available, by the way. You can download it. You saw from Reactome that there were Cytoscape files that you could pull into Cytoscape, but you can then change the visualization, change the network structure, all sorts of other things in Cytoscape. So what I'm going to do here is I'm going to click on File, Import, and I'm going to import a network from a file. So that file I just showed you with the source, interaction, target value, that's the sort of format that Cytoscape takes in. So I'm just going to because it's in downloads, isn't it? There we are. So I've got a file in here called test, So what it's got is the source gene, interaction value, target gene. And so I'm going to select the source. I put as a green dot here, there's the source node. I select the interaction as interaction, that's edge values, and the target as a target node. And I'll click OK. So it loads the network. Let's make it a bit more viewable. And so now I can see I've got a window here. It has all my genes, all the interactions. What I can do is change the layout. So I can scale this, zoom in, and I can then draw that out again. So it just gives me a bit of space between the nodes. And I can then reset that. So now I see I've got clear hubs going on here. So in here we've got AHR as a hub and DDoS as a hub. Stat3 and strap. Yep, so this is the starting point of a network with hubs, connections. What I want to do now is I want to I should add, yeah, save. What I need to do is I need to add some absolute values for the edges. I'll show you why in a second. So I'm just going to take that, I'm going to have an absint, and I'm just going to put the absolute value of, yes, okay, of this parameter here. I'll save that. Why I want the absolute values, I can use that to set the thickness of the lines. Obviously a line can't have a negative thickness, so I need to set that as an absolute value. So I'm going to close this session and start it again. So file, so I'm going to import the data network from file, no, not table, from file, console, file, import network from file. So now I'm going to set the source node as that, interaction value as fine, target node as the red one, and I've now got absint, which is the absolute interaction value, which will be used in the network. So I'm going to stretch that out as I showed before and zoom out. Now, what I need to do is, if I go to now a style tab, I can change the font size of my label. So I can set the default in this, I can set a mapping value, and I can set a bypass value. So the default applies it to everything. If I click on a particular node, I can set the bypass for that node and put a different size in. And if I set a mapping value, and I can map a range of values. Anyway, what I'm going to do is I'm going to put this as 21 point font. It still needs to be more, maybe put it as 40. Now I can see the size and the title of my nodes. Okay. Right. What I can also do, I can change the node shape here, and you can see that the hubs, and you can see that the hubs Okay. In a couple of minutes for tea, but I'll just show you this last bit. What I also want to do is, I'm going to minimize that. I want to set the width of the line. So I can select the value of absent as the width of the line, and I can select the mapping type as continuous. So what this does is allows me to set a range map. I can make the low value 0, the high value 30. I'm going to lower those a bit. And I'm going to apply that. So now you can see I've changed the thickness of the line depending on how much influence there was. So I can see this DDoS thing is really strongly connected, other things are less strongly connected. I can change the colour of the line as well. I'm going to use the interaction value, and I'm going to select continuous mapping, and that will allow me to show the stronger and weaker interaction values. So if I now minimize that, so you can see I can quite quickly build up interaction maps, highlighting things, highlighting edges, changing colors. If I want to move a node, I can just drag and drop it. Yep. If I want to see what a node is connected to, I can select first neighbors and directed. So now you see that's highlighted for me everything in that network that's connected to that bit. If I go to select nodes, invert node selection, I can now take everything out of that network that is not connected to that. So I can add in things, take things out, I can add tables, I can add extra values, I can add extra characterization. I can also take what's in this network and do an ontological search with things like Reactome. So I can say, I can highlight certain pathways that are appearing in this interaction map. So what hopefully you're seeing is that all these features, these tools, are starting to come together. They're starting to integrate, and you can move from one tool to another and just seamlessly transfer things through. So I'm going to stop on Cytoscape. We're going to have a cup of tea now, and then after probably a 10-minute tea break, I'll just quickly show you Kemble, and then we'll move on after that, and then we'll call it a day after that. It's all right, no noise. Okay, so I'm now going to talk a bit about Kemble. Everything you've seen so far, with the exception of maybe Pride, is a biological database, although Pride's proteins. Kemble is about chemistry, finding chemical things. So again, this is run through the EBI. If I click on Kemble, it's ChemBL. So I can use this as a search. So there's often highlights in here. I'm going to search though, and what I want to do is search for a biological target. So I'm just going to put EGFR for a start. Right. So this, sorry, it's taking a little while to search. Okay. So for EGFR, we've got 62 compounds been identified, 161 targets, 8,500 assays, and 959 documents. So what can I look for? Well, it's got papers here for a start, and I can see where these things have been published. It's got publications. I'm not so interested in publications. I want to find the molecule. So I'll click on compounds, tools, no, compounds, okay. So what it's now pulling up is 62 chemical compounds that are known to target EGFR. I've chosen EGFR because it's quite a rich space. It's just taking some time to load up. And I know there'll be a lot of things that might well target it. Sometimes you get less, sometimes you get more, but this is an obvious one. Sometimes there are other things we could target which are maybe we've identified, and we want to understand what chemistry we're finding around that. So great, we seem to have an error, but let's see where we get to. Okay, so we're starting to make some progress. I'm just waiting for some of these histograms to load. But basically, for each of these compounds, I can look up things like its chemical properties. I can look up its log P and log D values. So log P and log D are important for DMPK studies. And it's basically the log P, log D is how soluble, fat soluble or water soluble the compound is. So ideally we want a relatively low log P, log D. If it's insoluble, that's not much good. If it's fat soluble, it may have a longer retention in the body, which we also have to bear in mind as well. So we don't, some compounds will literally bind to fat and spend 20 years there, and that can cause other toxicity issues. We seem to be having some issues around loading things. Let's just dismiss that, see if that helps. So it helps me search for chemistry that might impact upon the biology. So you can see there's lots of inhibitors coming up here. IB is inhibitor, AB is monoclonal, or MAB is a monoclonal antibody. I'll just pick icotinib up and look at that as an example of what I can find. Hopefully it'll move a bit quicker because it's not searching for hundreds of interactions. So what I can get from here is immediately I can get a 2D structure for icotinib. I can get the structure. So what I said before is about these SMILES codes. So I can pull SMILES codes from Kemble. I could access this with an API and get hundreds of them, potentially. I have a standard INSHI and an INSHI key. That's another form of text that I could mine to look for phrases that are in that are associated with something. Sources, I can see where I've got this information from. Alternative forms, these are, if you like, sister compounds which are similar to that one. I've got features of the molecule. It's just loading, I think. Yep. So this is telling me whether it's a natural product, whether it's a chemical probe, whether it falls under the rule of 5, whether it's a small molecule, whether it's first in class, whether it's a chiral molecule, and whether it's a prodrug. So this is all about drug discovery again. I've got drug indications, so you can see here I put in EGFR. Well, EGFR is a known cancer gene, an oncogene. But you can see here it's coming up with neoplasm, lung cancer, non-small cell lung cancer, adenoma, carcinoma, bronchial neoplasm. And then what I've got here is it's the showing the mode of action. So it's an epidermal growth factor receptor inhibitor, which we found. Yeah. There's only one thing it seems to be linking to, but I can potentially start to find. Yeah, that's good. So I could, in some cases, it will show other genes here that it might interact with. So off-target effects, effectively. So I've got also here bioactivity. Let's see if I've got anything there. It doesn't tell me what those are. So under bioactivity, in some drugs I can get the IC50, which is the concentration that achieves 50% inhibition of growth or whatever parameter. So that's a useful feature. Let's see if that works. Activities. It's very slow today, unfortunately. Sorry.
So in this in this portal, we can search for a particular protein.
Yes, you can search for a protein if you know the.
Click on that chemical. It shows us the chemical drugs which interact with it.
Yes.
And the way they will interact.
Yeah, but that's not comprehensive. It's the ones so far that have been found to interact with it, because it is a database. It's not looking at new targets.
In this way, we can find drugs that we are looking for.
Yeah.
For a particular pathway.
Yeah.
Maybe it or.
If you know the molecule that you want to target,
Yes.
particularly if it's a human molecule, if you put it into here, you can probably find compounds that will potentially.
Is it kind of targeted approach, we can say?
Yes, yeah.
Okay, thank you.
So where we're getting to, we've shown you how to analyze data, we've shown you how to find targets, we've shown you how to find targets that are central and influential. If you take those targets that are central and influential, you can potentially identify small molecules from this that have the that could be drugs that interact with that target. This is a database-based approach, though. You could also use docking, convolutional neural networks, other techniques.
So most of the databases that we have talked about are more reliable on human data, human genome data.
Most of them are, yes, yeah. But Kemble isn't just human data. It has agrochemicals in there, for example.
Okay. So is there any other data set which includes more about plants and other species?
The trouble is everybody focuses on humans and then plants come second, yeah.
If you have any idea about it, please share with us.
Well, Geo has got plant data in it. Geo. And then this will have some plant data in it because it's got agrochemicals in it. And if you put the plant-based gene or protein that you want to target, it should be able to see whether there's anything in there.
I tried doing that and it did show, but the data it has driven from is a Homo sapien one.
Yeah, but at the end of the day, if it's showing activity on a human gene, it's potentially indicating if an analogous or homologous plant gene that would have the same activity on.
The protein is same, the effect will be same.
Yeah, the protein, it's going to be similar. You can't say it's going to be the same. But you could try it out. That's the thing. It's giving an indication. Until you've validated it, it's still only in a database. So, yeah. Okay. No worries. Sorry, did you have a...
First answer is...
So you haven't found anything?
Actually, the compound I'm looking for in PathChem is something else, but in Kemble it's not.
If you can find the Inchi key or the Smiles key, you can search on that in Kemble.
I can search using the Smiles key.
Yeah. That's probably more accurate than using the chemical name. So, yeah. So what I've got here is yes. So X phase, it's not complete by any stretch of the imagination, but P. Kemble, biolabel, primates, taxonomy. So what I have to do for the log P and the log D value is, if I look for EGFR, then it will give me a table, if it loads, because it's not loaded so far. It'll give me a table of compounds and their log D and log P values. It doesn't have it for everything, and often it's a simulated log P and a log D based upon other things. Let me see. Type histogram, find term. Let's see if we can find a term. Here we go. This is. So if you see here, look, I've looked at everything that hits EGFR. And then, so I've got the drug phase, which is, okay, that's useful. I've got RO5 violations. From memory, these are toxicity issues. So an RO5 violation is a flagging of a toxic compound. Yep, so we have to be careful of that. Molecular weight, yeah. Here we have the log P. So you see I've got 12 things which have a log P of between 5 and 5.7, 23 things between 4 and 5, and so you can see I can, then if I click on that, I can pull compounds up within that group. I've got mainly anti-neoplastic and immunomodulatory. Here I've got the number of targets. So, for example, if I click here, these are compounds which have multiple targets. So those have more risk of potential toxicity because they're hitting other things. But if I want to model, let's come up with papers again. click on compounds.
What's the other way around? Like if you put the name of the drug in the search bar, can it show how many pathways or like...
It won't show pathways, but it will show known molecular targets and proteins or genes that interact with that. So my view of toxicity is if I find a compound based upon a pathway, then I can then take the interactors of that, put those into network inference, find, if we like, a representation of the toxicity of the compound, and then find the drivers of toxicity. So I can build a simulated tox model based upon the known interactors with a target compound. I could use Pathway Miner to then fill the gaps in that as well, and start to understand what's happening around that space in terms of toxicity.
Then also we can, like if we come to know regarding that, we can search for something which can cure that toxicity.
Yeah, so you might find a co-molecule that is ameliorating that toxicity.
Yes.
Yeah. Yeah. So that's pretty much Kemble. There's a lot to go at for Kemble, and I could probably spend the next 3 hours talking about it. But again, have a play, have a look, see what you can find there, see what data you can get out of it. It's all part of the toolkit that we're trying to show you for this workshop. Okay? Any other questions? No? So, okay, well, I'm going to stop now. I've got to get back because we've got this panel discussion this evening, so I need to get back for that. But thank you for coming today, and I'll see you tomorrow. Thank you.